Thank you. Welcome, everybody. We are really excited to start this session now at 11 o'clock on the cumulative anthropogenic impacts on key Arctic species. There are 4 conveners of this session. Nadja K.M. here with me, Ben Laurel also with me, Frans Muter could unfortunately not be here, and myself, Frode Vikebø. We've already had a great talk this morning by Katrine Burger, and we will continue with now a talk pre-recorded by... It's still off, okay. Ja, ja.
Har ikke laget til rommet.
Ja, ok. Jeg kan snakke ekstra høyt.
Ja, nei, men da blir det ingen lyd på videoen.
Ja, ok. Er den ok?
Ja, nå hører vi.
Okay. So we will do it again so that also people that sees this through streaming will also be able to hear. So welcome to this session on the cumulative anthropogenic impacts on key Arctic species. Co-conveners Nadja K.M, Ben Laurel, and also Frans Muter, who could unfortunately not be here, and myself, Frode Vikebø. We've had a great talk this morning by Katrine Burger, and we will now have a pre-recorded talk by Nadja Steiner. And I would also like to tell the next speakers that halfway through the presentation in advance of you need to go down and get your microphone set. You should look at this screen down here so that the people viewing from afar can pay attention to your talk. And also, please stay on the podium until you've answered questions. And please pay attention to the clock and keep your time. So we will start with the talk by Nadja Steiner.
Good morning, my name is Nadja Steiner. I'm a research scientist with the Department of Fisheries and Oceans in Canada, and I would like to thank the organizers for inviting me to this session. I apologize that I cannot be there in person. I would like to acknowledge that I'm speaking to you from the traditional territory of the oceanic people in the west coast of Canada. And I would like to acknowledge also my collaborators Tessa Su, Kathy Reeder, Patrick Farnell, Antoine Haddon, the community of Olokakdok, and the BEPSI research community. Today I'll be speaking to you about climate change impacts on marine ecosystems and ecosystem services in the Arctic. I'll give you an introduction, talk a little bit about climate change and environmental drivers, sea ice ecosystem services, connecting climate model downscaling with communities, and a little bit about marine conservation. In the IPBES IPCC report on biodiversity and climate change, we mapped the climate ecosystem people connection in the Arctic onto the IPBES conceptual framework. The conceptual framework highlights how direct drivers, both natural and anthropogenic, can impact the good quality of life of Arctic people either directly or through their impacts on nature and nature's contribution to people. In addition, good quality of life can be impacted through institutions and governance, both on international, national, and regional levels, as well as through the anthropogenic assets that are available to the Arctic peoples. In the coming slides, I will talk a little bit about some of those sections in more detail. I would like to start with the environmental drivers, which through climate change impact the marine ecosystems. To do that, we evaluated the coupled model in the comparison project CMAP6 with respect to marine ecosystem stressors in the Arctic. We used 11 Earth system models. We looked at ecosystem stressors: temperature, ocean acidification, oxygen, and sea ice. We looked at 3 different depth ranges, as well as divided the Arctic into 14 separate regions. What is shown here are the multi-model means over all Earth system models, and we are looking at the regional differences as color-coded by this map down here. We're looking at temperature in the top 50 meter, sea ice concentration in summer, and sea ice concentration in winter. Unsurprisingly, we're seeing different initial states across regions, but we're also seeing consistent temperature increases. The warmer temperatures are for the more subpolar regions. For sea ice in summer, we see all regions are moving towards ice-free summers by 2080, so the pace is different across region. And for the winter sea ice, we initially still see fairly consistent ice cover in several of the more northerly regions, but then we see a fairly abrupt decrease even in winter starting in about 2014. For ocean acidification, we are looking at the calcium carbonate saturation state. Calcium carbonate in form of aragonite and calcite is formed by marine organisms to form their skeletons. If the saturation state goes close to 1, they have difficulties in forming their skeletons, and if it gets to 1 or even below, they even start to dissolve. In the Arctic, we see a lot of regional differences again, with most advanced ocean acidification in the Siberian Laptev Sea, as we can see here, but also in the Western Canadian Arctic. And there we have already seen in observations that pteropods, which are formed from aragonite, starting signs of dissolving. For the high emission scenario SSP 585, all Arctic regions show aragonite saturation, under saturation by 2080. When looking at trends, it is pretty obvious that we cannot approximate the whole time period from 1980 to 2100 with one linear trend. However, if we split the time period into blocks of 40 years for early, mid, and late century, then we can reasonably approximate each block with a linear trend. And this is what we have been doing here for temperature in the top 50 meters. We see here the early century, mid-century, and late century. On the x-axis, we're seeing the regions indicated by number, as shown here. And the different colors indicate the various Earth system models. So what we're seeing here is in black the multi-model mean and standard deviation, and that for the 3 time periods. And what we can see is that the models show distinct regional differences in trends, as well as a large spread among the models. The trends in black as well as the model spread are increasing over time, with the last time period showing significant difference among the models in the trends. Here we're looking at the same thing for omega aragonite. Again, the 3 time periods, early, mid, and late century. The saturation state decreases with increasing CO2, which is triggered both by the increased PCO2 in the atmosphere and then with the retreat of sea ice that allows more uptake in the region. But it is increasing with temperature. So what we're seeing here is that when we move from the early century to mid-century, that we see a strong increase in the trend, like a stronger negative trend. But then by late century, this trend is actually becoming lower again. And that's because the mid-century is the time period when we see the strongest change in sea ice, and we also see the start of strong winter sea ice degrees in several of the regions. In addition to the change in trend becoming lower, we also see that over time for ocean acidification or omega aragonite, the spread of the models is actually decreasing compared to the temperature where it is increasing. And this indicates that there's a limit in uptake capacity in the ocean. One other thing I would like to point out here is that we are using CANI SM5 for 2 versions, 2 different biogeochemical models, and what we can see here is that both those models indicated with the red solid and red open circle show quite large differences in some of the regions. And essentially what that highlights is that the differences that we are seeing among the models can be both through the physical differences between the models, but also due to differences in biogeochemical models. Staying with ocean acidification and omega aragonite, I would like to look at 2 different scenarios. The high emission scenario, SSP 585, indicated in red, and the lower emission scenario, SSP 245, indicated in blue. We're looking at the individual models in thin lines and multi-model mean in thick lines. And then we're looking at 3 different regions, the North Beaufort Sea, the East Siberian Sea, and the Barents Sea, right here. So what we can see is that ocean acidification or saturation state until about 2035 is independent of the scenario. Afterwards, the higher emission scenarios show faster ocean acidification progression, as indicated in red. For regions with most advanced acidification, like the East Siberian Sea and the Beaufort Sea, aragonite undersaturation will be reached in all scenarios. For other regions, such as the Barents Sea, the regions with lower emissions can avoid undersaturation, as we can indicate here. However, the annual mean is not necessarily the most relevant or the only relevant for marine species. So we're looking here at the annual cycle for temperature in the top 50 meters. We look again at the 3 different regions, and what we're seeing here are the individual model in colors, and in black we're seeing the multi-model mean, and we're looking at the mean for 1981 to 2000 and then for 2081 to 2100. And what we can see is that we see strong change or increase in the seasonal cycle for all the models, and we also see that in all the regions. In some of the regions we go from like almost no existing cycle to a very prevalent seasonal cycle. And in the bottom figure, we see like the same thing for all the regions and we're looking at 3 time periods, including the middle 2031 to 2050, to see how over time the mean, just indicated with a dot, and then the annual range indicated by the bar is changing over time. And we see that across the board in all the regions. To better describe the value of ecosystem to humans, the description of ecosystem services has been introduced. Ecosystem services are divided into 4 categories: habitat and supporting, provisioning, cultural, and regulating. The research community BEPSI has prepared a thorough review of climate change impacts on sea ice ecosystems and ecosystem services. Those are summarized in a policy brief, and I will get in a little bit more detail in the following. Either directly or indirectly, sea ice contributes to regulating services through control of planetary radiative balance via the production of biogenic aerosols, atmospheric cleansing, and the release or uptake of climate-relevant gases such as carbon dioxide and dimethyl sulfide. Sea ice provides supporting services in form of habitat, including feeding grounds and nurseries for microbes, meiofauna, fish, birds, and mammals, as well as life cycle maintenance. On the left-hand side, we see a selection of species that depend on sea ice, and I apologize, those include both Arctic and Antarctic species. And on the right-hand side, we see an example of energy transfers from sea ice algae through Arctic cod to higher trophic levels. Sea ice provisioning services are provided through harvesting of country foods and the supply of potential medicinal products and genetic resources. Cultural services include less tangible components such as the inspiration and attraction for cultural activities, tourism, as well as research, and the base for indigenous and local knowledge systems, cultural identity, and spirituality. All these sea ice ecosystem services respond to changes in the underlying sea ice ecosystem. The BEPSI community compiled a synthesis of future changes in Arctic sea ice biogeochemistry and ice-associated ecosystems. Divided for the Eastern Arctic, Central Basin, and Western Arctic, we looked at changes in the physical variables, biology, and chemistry to see if they are either likely positive, possibly decreasing, or possibly increasing, and so on. In results, we find that most of the sea ice components are likely to decrease, like first-year ice, thickness, and extent, as well as snow, but we see increases in rain, melt ponds, and leads, as well as light. For biology, most of the components are decreasing: habitat, biological carbon pump, sympagic fauna, megafauna, and biodiversity. And things are very similar across the Arctic. The chemical components show mostly a possible increase. Research is not is divided in terms of what the possible increase or decrease is for nutrients, primary production, and the carbon sink. Connecting climate model downscaling with Inuit community impacts. To communicate climate change with two Inuit communities which are most impacted by climate change is not trivial. The time series type of graphics that we usually use as researchers don't resonate with the affected Inuit communities. They are not really familiar with this type of presentation. So we have to think about methods that are possibly closer to their knowledge system and their familiarity. What we're seeing here is a schematic out of the Inuktitut curriculum in the Inuvialuit settlement region in the Western Canadian Arctic. It essentially shows that the cycle of season is tightly linked with the cycle of life and the circle of belonging. The students revisit this schematic throughout their elementary school years, middle school years, and then again in high school years. So this is something that they're intricately familiar with. So what we are trying to do is to build an Inuit calendar about the seasons and try to link that one with the climate modeling, with the idea behind it that we know that climate change impacts the cycle of season, and if we better understand how the climate change causes these ripples and how they feed back to the cycle of life, then hopefully it will help them to better understand how climate change will impact them in the future. So in the community of Olokaktok in the Western Canadian Arctic, in collaboration with the Olokaktok community members led by Alan Pogotak and the Olokaktumit Hunters and Trappers Committee, we are working on the development of a traditional calendar for subsistence harvesting. Essentially, this calendar contains information on the appearance of species and the associated harvesting activities across the season. And we specifically focus on identifying what are the environmental indicators that indicate a change or a difference in harvesting activity, and how have those changed over time. To do that, we had multiple visits in the community and are still actively doing that. We've met with elders and hunters, language teachers, students, and had multiple reiterations of this calendar. And as you can see in this example, like we came with one draft and then we wrote all kinds of additional things on it and are continuing to work on that. And the idea is really for the community, develop a tool that they can possibly use, but also for us to develop a method that can then be applied for other regions or other locations as well. To link the calendar with environmental modeling, we're starting with the example of sea ice. We're looking at a high-resolution model in the vicinity of the community of Olokaktok. We average over a region of the Amundsen Gulf, looking at the ice concentration for some example years that show very low ice cover in the summer, or very high ice cover in the summer, low and medium ice cover. And then the idea is to look at what differs in the calendar in a low ice year versus a normal or a high ice year. And this can then be used for future climate projections, but also for seasonal predictions. This example here shows the number of months without ice in the Amundsen Gulf. In past simulations, we really see a lot of variability with years where there is no ice, no months without ice at all, but up to 3 months without ice. So there's a lot of variability, and that's where it really becomes important to look at seasonal prediction that will then help the community earlier on in the year to identify, or is this a year where we have to expect the whales early or late? Will it be a good seal year or a good char year also, and so on? But also to identify in the future that something that has been a usual exception is now suddenly the new normal. Of course, we are not limited to sea ice only. We can also look at other variables such as temperature, primary production, ocean current, stratification. And we can use available time series of body condition or harvesting data for particular subsistence species and see if we can use the model to provide any linkages between those. But what I'm particularly interested in is the changes in ice algae to pelagic production over time or with a changing climate, because these are directly linked to the forage species that then feed the subsistence species such as ringed seals, beluga, char, and bowhead whales. And what we can see here is that an example for Arctic cod, where young Arctic cod feed on the ice algae. But if this habitat is disappearing or becoming less or coming at a different time, it might impact other species that are then coming in or replace Arctic cod, which then in turn feeds back to the subsistence species. Our ocean downscaling model includes both sea ice algae as well as ocean primary production. And in this example, I'm showing simulated changes in sea ice algae 2056 to 2080 minus 1980 to 2009, so 30-year time periods in the recent past and in the future. And we're seeing that overall this is for spring. We're seeing areas where we see a decrease in ice algae production, but also some areas where we expect a positive change in ice algae production. And we're looking at the same time for those regions at changes in the least growth limitation factor, which essentially means that there is a release in the limitation of growth, which is mostly through light in this case, and but also changes in the growth period. And what we find is that for the regions where we have like an increase in growth, it's mostly due to a combination of a longer growth period, but also of a release of that limitation factor. When we look at the same figure for fall, which is like usually a lower production bloom, but it's still a relevant bloom in the region, the model suggests that we mostly see a reduction or a loss of that fall bloom in the future. I wanted to have like a quick glimpse into marine conservation and marine protected areas as well. Marine protected areas are key management tools to maintain biodiversity, ecosystem function, and to secure the delivery of marine ecosystem services. As we indicated before, like the sea ice ecosystem supports all 4 ecosystem services: provisioning, regulating, habitat, and cultural. For the identification of marine protected areas, the designation of ecologically or biologically significant marine areas, or EPSAs, is a key step. To aid in the identification of MPAs in the Arctic, we performed an assessment of the sea ice ecosystem against the 7 criteria for EPSAs. The 7 criteria are uniqueness, special importance for life history stages, important for threatened, endangered, or declining species, vulnerability, biological productivity and diversity, and naturalness. We assessed the sea ice ecosystem as high for or medium for all of those criteria, which strongly suggests that the sea ice ecosystem should be included in the identification of potential marine protected areas. Including targets for marine conservation into the Sustainable Biodiversity Goal SDG 14, Life Below Water, has encouraged countries to move forward in designating marine protected areas, including in the Arctic. At the 5th International Marine Protected Area Congress in Vancouver in February 2023, it was called for accelerated progress on that SDG 14, but also to increase efforts to achieve Target 3 of the Global Biodiversity Framework, which is to conserve 30% of marine and coastal areas by 2030. And that there is a particular focus on partnership with Indigenous peoples to enable Indigenous-led conservation. On the right-hand side, there is a summary of conservation areas in the Arctic as they are currently either worked on or identified. This includes the Tuvalu-Ituk MPA in Canada, which is the first or the only MPA that has been designated specifically due to its sea ice-related ecosystems. There's other conservation efforts with key indigenous involvement, the Tulariop-Imanga National Marine Conservation Area, but also the first indigenous protected area in the in the Hudson Bay. So there's several processes ongoing or recently established, but there's still lots to do, and we hope that the work in sea ice ecosystem services that we highlighted will help in some way to contribute to that. In summary, the Arctic marine ecosystems are experiencing enhanced stressors with regional differences and varying biases over time and space among models. Trends get faster paced over time, and annual cycle amplitudes are increasing. The sea ice ecosystems meet the criteria for EPSAS. They support all 4 ecosystem services categories and deserve specific attention in the evaluation of marine protected area planning. Global emissions driving climate change are directly linked to the demise of sea ice ecosystems and its ecosystem services. The response of sea ice-associated primary production by geochemical processes is regionally variable. However, the effect on ice-associated mammals and birds are predominantly negative, subsequently impacting human harvesting and cultural services. The key mitigation measure remains a reduction in carbon emission. Okay, that's all for me, and I will leave you with a little fun drawing of the ecosystem services for the Frontiers of Young Minds journal, and I'm available for questions.
So my understanding is that we are not able to interact with Nadja after this presentation, but we can contact her by this email address and follow up. I would also like to note that we have one gap in the schedule for today. There's a gap between 2:15 until 2:30, and we will use that gap for further discussion. So please just make notes and bring them up at that point. Still, we have 4 minutes until the next presentation, so we can open up for some immediate reflections based on these presentations. Would anyone like to comment? Don't be shy. One reflection is that I think there was a really interesting approach to make this life cycle gap, the way of life, and connect that to the needs that we have from these climate model projections. But how to do that for multiple and adverse groups, that would be an interesting thing to follow up. And probably Nadja and colleagues have already done that. Other comments?
Yes, thank you. I think that's kind of a challenge that we have for all types of communities to link impacts to impacts on the ecosystems to further to ecosystem services and consequences for society. So that is a general challenge, and not only for some communities.
Definitely. Samuel?
Yeah, and I think it's so important to engage those communities because that's where we can also look at adaptation or mitigation issues, which obviously have become increasingly important.
Also, there was a really nice assessment here of multiple climate models across the Arctic. And I assume that many here would like to know what were the details for their particular pet region. So the downscaling effort is also something that we would like to know more about. Any further comments before we start the next presentation? So I think that Hjálmar, you can simply come up here and we will start in about a minute.
Thank you. Am I on? Yeah.
Yeah, very good. And
I have to stand there.
Yeah, you have to stand on the podium. And please look at the screen down here, because then it will be easier for the people who follow.
Sure.
And you will have to wait in 1 little minute so that we follow the schedule. But the talk today by Hjálmar Hátún is the physical drivers of biogeographical shifts in the northeastern Atlantic and adjacent seas. And as I've said, please make notes and bring them up during the discussion if you cannot address all with questions immediately after the presentations. So the floor is yours.
Thank you. Yeah, I'm Hjálmar Hátún. I'm from the Faroe Marine Research Institute in Faroes. And this title is actually a part of a research topic in Frontiers in Marine Science. You see the logo here. Red currents go north, they're warm, and they have a fish tail, pelagic fish. Blue currents come south, and that's not a carrot, it's Calanus. And then you have this subpolar gyre, which revolves around there, and my work revolves much around this one, but physical drivers of this region. I was one of the editors, Teresa from Iceland, Øystein from here, and Peter from Denmark. And this resulted in 19 papers with more than 60 authors. So this is a kind of a sales pitch for this one to lure you to read it. Much revolves around this one. It's kind of been my trademark. This cooling and convection fills up this subpolar gyre, cold and low salinity, rich in nutrients, waters that revolve counterclockwise, south of Greenland and Iceland. And it's north of the Gulf Stream and a subtropical gyre, warm and saline waters. And we've shown that the marine climate here in our waters here is much governed by this gyre dynamics. When this gyre is strong, it's subarctic, its salinities are low, temperatures are low, it's prosperous on these boundary shelves. In the mid-90s, and it can do this in between, and then it changes a lot. It opens up this window for this northward protrusion of subarctic waters, and salinities go up and temperatures, and it changes the entire ecosystem. So strong subpolar gyre, salinities are low, but this can happen also. And then we came up with say that this is the Atlantic inflow that go towards the high Arctic, into the Norwegian Trench, also into the central parts of the Nordic Seas, and towards the Labrador Sea and north of Iceland, called the Atlantic inflow. And then we made this gyre index for this dynamics, and strong is down there. So in the mid-90s, this subpolar gyre was really strong, and then it weakened suddenly in the mid-90s. And then we see salinity west of the British Isles and close to Iceland, because it's a conservative tracer of water masses. And that's what really matters, these changes of water masses. And to look, it was really strong, and then salinities were really low here in the, as I mentioned, the early 90s, and then the weekend, and then we got record high temperatures and salinities. Yes, this is not up to date, but I'll continue the story. And to see how this looks in space, this early 90s period, we see this gyre really protruding towards Europe. And then a few years afterwards, it weakened, and we had a major change of water masses and everything in these waters. We can have these major shifts. We can also have more intermittent salinity anomalies. When salt goes down, it's something like this, and this kind of blows life to our shelf ecosystems. And then in this topic, research topic, we are just calling physics and nutrients as the zeroth trophic level. And then we come to first is phytoplankton, second zooplankton, third is fish, third and fourth is seabirds, and the top predators are the fourth trophic level. And I'll refer to this. And after this, I'll talk fast, so if you don't get anything, just know this. It came out as an ebook, almost 300 pages of this biophysical linkages in this part of the world. So those people or everybody, if you want to get updated, this could be one source. And we have this editorial here, and that's the offspring for what I'm talking about right now. And of course, it's not easy to compile all of this into 15 minutes. So I just structured according to recurrent aspects in all these papers. One is phenology, timing of things. Spatial segregation and connectivity. And then if it's connectivity, then maybe predictability. And some biological hotspots where much happens. Major shifts, you can call it regime shift or tipping points, I don't care, they're just big changes. And then some long-term trends that could maybe be climate change. So on the backdrop of this image by Ofstad et al., we see the main biomes or ecoregions or biogeographical zones, or what you want to call it. The boreal, very rich one here, and the subarctic, and then the shelf biomes. And then I'll just go, and this goes fast, sorry for this, but to take these papers and trophic level 1, that's phytoplankton, and now it's phenology. So Richard and al, they look and see big changes or differences between the subarctic and the subpolar gyre and these different regions. This is a study by Homland et al. It is trophic 3. It is spring spawning herring in the region. And they are feeding much more extensively and for longer into the season, so it's change in phenology towards Iceland. Here's a study from south of Norway, coastal station here. This is the main zooplankton, Calanus phenmarchicus and Calanus helgolandicus. They are shifting forward in time, and also this warmer water species now is getting a spring peak. It didn't used to do so after 2002. And here's a study on phytoplankton and remote sensing for Barents, Norwegian Sea, and the North Seas. They see changes also in the matrices for the spring bloom and the summer blooms. Then we come to segregation. The reason we have these biomes is, of course, we have fronts, and so things are not just flowing easily across, so that's kind of segregation. These two studies, so now I'll not show them once more, I just show them by reference. Other studies, this is one based on core drilling in the seafloor across the Atlantic. It is plankton, and I see differences in the boreal and the subpolar gyre and in the Labrador Sea, naturally. So that's a type of segregation. This Miesner et al., it is physics and fish. The fish is this pelagic fish stock, blue whiting, and they're very picky where they spawn. They don't like this bluish cold water, so they get limited to the European slope. So it's kind of segregation down there. And also here this is a study on anglerfish. And of course, these water masses are not just two-dimensional, they are three-dimensional, so there's also a vertical aspect to segregation. This species is boreal, so it wants to be on the warmer side of things, but close to the cold one in the vertical. And also we have this zooplankton river coming in this the Iceland current in here. And there's a very strong frontier and strong segregation between the zooplankton community there and there. So physics and zooplankton and fish. The fish is herring. And then oppositely to segregation, it's connectivity. Following these Atlantic inflows, they are connecting all the way from here, all the way to the high Arctic. And if there is this predictable overflow, it will be advective lack, and if lack, there's memory and maybe predictability. The study on blue whiting, they find a predictability potential down there of maybe 1 year. And this study up in the Barents Sea, physics, phytoplankton and fish, also model study, they find 3 or 4 more years up to the Barents Sea. So altogether maybe a predictability horizon of 3 to 5 years. And connectivity is not only in the surface, because these, as we know, the water masses are three-dimensional, so these waters, they are sliding underneath the others. So they might actually connect to Norway, as actually Nansen said more than 100 years ago, into this channel between the Faroes and Shetland, the Faroe-Shetland channel, and also maybe into the Norwegian Trench, all the way down south here. So that's a kind of connectivity in 3 dimensions, which Kristen J is looking at. When we have more Arctic water, we see more of, for example, Calanus hyperboreus, a species that's endemic to the cold waters. And we also see connectivity into this, it's called the overflow, this is the deeper limb of the AMOC, overturning. And also you can see these species actually re-emerge all the way down south there, so they could actually feed onto the Norwegian shelf as well. Then the hotspots. This is one. Blue whiting are really congregated when they spawn, so that's a hotspot for biological activity. Another one is on the western side of the Faroes. That's where oceanic waters go onto the shelf. And this anglerfish, they love to spawn right there. So that's one hotspot. And the zooplankton hotspot is also drawing herring, so that's a feeding hotspot for herring. And also in this region, this is also where we see by far the largest egg production of Calanus phenmarchicus. So it flows this oceanic species, and they really produce eggs there. Another hotspot, and this study just underlines what they say. also herring, that this is really a feeding hotspot of herring and blue whiting and mackerel. So huge biomasses in this region of pelagic fish. Then the shifts, let's call them regime shifts. So these are temperatures from the study down here. And I showed this strong gyre, SPG, subpolar gyre, and a weaker one I stopped in 5, but something has happened afterwards. So in the mid-90s, I would say, is so big change as we could call the regime shift. And then we were up here, but we have gone kind of down again after 15. And if you look north of this community here, this is small, I know, but these are stages of Calanus marcus. When it's bluish, it's larger stages, and also hypoboreals, and that's kind of signature of a subarctic state. While in between we had Atlantic state in this region there. That changed around 2003. And then this more subarctic started after 16 again. What happened there was, maybe you heard about this blue hole in the North Atlantic. Some people associated with AMOC. That is not true. It's this kind of dynamics. The subpolar gyre expanded, and so did this East Atlantic current with these subarctic waters in towards Norway. Also described by these two papers, and also influencing the distribution of feeding herring. And also you see this, when it went down, then you see changes all the way down south here also. And also for this anglerfish here, they like the Atlantic, so they had really a peak when it was Atlantic. So it changes the community. And now another study also in this one, of course, it's Atlantic salmon for these waters there. And when this happened in 2003, it was not good news for salmon either. They also had worse times. I've heard some rumors that they might be doing better now. I don't know if that's true. Some people here in Bergen last year said so, but really interested in knowing reactions to this one. And here's a study on this clam called Arctica islandica as a proxy for production. You got really long time series, and it's the same story, 2003, not so productive. And then last one, trends. This study on the Calanus egg production has been going down, unfortunately. And so has also the production and the fish stocks and so on the fair shelf. So our benthic system is really doing worse and worse, unfortunately. Long-term trends also in the feeding pattern of herring, in the primary production, this region, and also in the zooplankton community down here, more Calanus helgolandicus. And the study, this is the final one I'll talk about, is ocean color imagery. It's phytoplankton in Svalbard fjords. They are apparently getting darker and darker. So sorry for, I know it was a lot. But if you're more curious, you can just take a picture of this one. And now to the most important for me here is that following along these lines, now in August, late August, we're going to have this PhD course in the Faroes, at our institute and the university also, and 2 days on board our brand new research vessel. It's going to be super exciting. We just got this program funded by Denmark, where we have 6 projects coming up. So we want to kind of create coherence around that project, but also to be open to researchers from abroad. So please, if you're a student, or if you have students, have them write to this one. Thank you.
Thank you, Hjálmar. So we have time for one question at least before we move to the next speaker. Use this microphone. Ben?
This might be a naive question, not a physical oceanographer, but what is the state of knowledge as far as doing forecasting the state of the subpolar gyre? Is it something that's able to be forecasted out in likelihood from one year to the next, or?
People have studied that. I think Cachaloma was the first one in 2009 to do so, with model studies. So, yes, if you look at these forecast models, the greatest predictability potential in the world oceans are, in addition to ENSO region, these waters, because they involve really a deep water column, 1-2 kilometers. So you have lots of thermal inertia memory in the system. So her conclusion was that when the system is in an extreme situation, sometimes it's predictable when it's really in a state, and then changes happen to the atmosphere, they can predict what happens. But if you are kind of in an average state, then it's not so predictable. So predictability is not just something that goes constant all the time. Sometimes the system is more prone or more potential for predictability.
Quick question, quick answer.
Yeah, well, it's very comprehensive work, Hjálmar, so thank you. I was wondering, one of the consequences of the regime shift in the mid-90s was that there was a quadruple of the recruitment in the blue whiting in a 10-year period. And this is the biggest resource in the North Atlantic, in terms of biomass. The last 2 years has been the strongest recruitment of the blue whiting on record, even stronger than what we witnessed in the mid-90s to 2000s. So how is there a regime shift ongoing? That's my question.
I'm super happy you asked that question. I'll try to make it fast. One of the projects here is actually to try to predict recruitment to blue whiting. It's not just the gyre, it's what is Winstra's curl we find as the most likely driver west of the British Isles. And actually last year I said there's going to be really a peak in blue whiting recruitment. And this fall when they had the working group, boom, there it was. So, and actually many years back in the ICES Decadal Symposium in Santander, I played with this idea. And it has held since. So those changes are more erratic. If that's just a regime shift or more erratic changes in the atmosphere, I think Winstra's curve on blue whiting recruitment. So one of these projects is on that.
Thank you, and continue discussions afterwards. So the next speaker will be Kristen Sora on the influence of historical climate drivers on the Beaufort Sea shelf marine food web. Take it away.
Thank you for having me here today. So today I'll be presenting some of the preliminary results from my second data chapter of my PhD thesis. So first I would like to begin by acknowledging that the land on which I reside is the unceded territory of the Coast Salish people, and this includes the territories of the Musqueam, Squamish, and the Tsleil-Waututh nations. And I would encourage everyone to learn about the native land on which you inhabit at native-land.ca. And I'd also like to acknowledge all of my co-authors as well for this. So climate change impacts on marine ecosystems are particularly acute in the Arctic, where changes in recent decades are amongst the fastest in the ocean, raising concerns for conservation of key ecologically and culturally significant species. Multiple climate-related drivers affect marine species survival, altering ecosystem productivity, food webs, biodiversity, and fisheries. And these include increases in sea surface temperature, losses of sea ice, and decreases in dissolved oxygen concentration and changing ocean salinity. So within the Arctic, we have the Beaufort Sea, and shown here is the Canadian portion of the Beaufort Sea. In purple is the Inuvialuit settlement region, and the Beaufort Sea is one of 5 priority areas for integrated management, indicating that there should be a precautionary approach to sustainable use, development and protection of areas and resources, and incorporation of social, cultural, and economic values. And the Beaufort Sea encompasses diverse ecosystems such as the Mackenzie Delta estuary, deep basin features such as the Beaufort Gyre and the Beaufort Sea Shelf, which is our focus today. So here I have a zoomed in map of the Beaufort Sea Shelf, and which is my study area for my thesis. And it has depths from 20 meters to 500 meters depths, shown here in blue. And we also have the TNMPA, which has the 3 sub-areas here in green. And it was designated in 2010 with the management objectives to conserve beluga whales, anadromous fish, and birds, along with their habitat, ecosystem, and traditional harvest. And there's 3 communities that harvest the area, including in the TNMPA. We also have the map here of the transects of the Beaufort Regional Environmental Assessment Offshore Marine Fishes Project. It's the most recent and most comprehensive assessment of the Beaufort Shelf, and data from these sampling stations and transects were used to update the fish groups in the Beaufort Sea Shelf ecopath model. So my current research applies ecological modeling approaches to understand how existing conservation measures, such as protected areas, can support adaptation and resilience to climate impacts on the Canadian Beaufort Sea ecosystem. So first, we updated the existing ecopath model from my first chapter, other previously published studies and projects, And the models developed in this study are used to provide a baseline to investigate the impacts of climate change onto the Beaufort Sea and projected to 2100. And then we investigate the environmental drivers, including temperature, salinity, and dissolved oxygen on these ecological dynamics and the long-term health and key species. And then we explored the climate impacts onto the trophic levels, biomass, and diversity of this ecosystem. So for my thesis, we used ecological modeling tool EcoPath with EcoSim. It's a useful tool to describe the complexity of the ecosystem and the impacts of environmental or anthropogenic changes onto the ecological groups defined in the model. EcoPath is a static snapshot of the ecological model, and shown here we have the EcoPath flow diagram with trophic levels on the y-axis. And so down here we have it from producers all the way up to the fisheries group. And so then the size of the squares are the relative biomass of each group, and the gray lines are their connections to one another. And so shown here I have highlighted some of the key species we'll be focusing on today, so beluga whales and Arctic and polar cods. And then EcoSim is the time dynamic model that builds upon the initial EcoPath model. It's fit to time series, so for this project it's from 1970 to 2021, and it's used to address ecosystem-based management questions. And for this chapter of my thesis, we use species ecological niche and climate forcing, and from these previously described environmental drivers. And so to calculate this ecological niche, we combined the global occurrence data of the species. And for example, here we have the distribution of Arctic cod. And then we combined it with the global model data of these climate variables we have here. And so that gave us the climate threshold for each species shown here. And so that was for each species and for each climate variable. So then the time series climate variables from this study were obtained either from observational data sets or from the model data sets, and that was from the Earth system models, the GFDL and the IPSL. And then we had the two scenarios we compared as well, so that's the 2.6, the low emission pathway, and the 8.5, which is the high emission pathway. And so to have a complete sensitivity analysis, we ended up creating 13 separate models. And so the first was this baseline scenario, and this was determined by the previous studies we had conducted, and this is using the observational data of temperature and sea ice. Whereas these 12 other models were a combination of the temperature and sea ice, dissolved oxygen, and salinity. So for example, the one that we have called GFDL 2.6 oxygen only is a combination of temperature and sea ice from the observed data, and then the dissolved oxygen from the GFDL 2.6 modeled values. Similarly for salinity, and then finally for all parameters, it's a combination of all of the climate variables. And so, the same structure then was then used for all of our other Earth system model values. So I'll now start with some of the preliminary results that we have from this thesis chapter, and we'll just be focusing on beluga whales and Arctic and polar cods today. So first are these changes in trophic levels, where we have the trophic level on the y-axis and our years on the x-axis, and so again, that's from 1970 to 2021. And then also the lines that will be showing up shortly are from these 13 models that I just described, and they are the mean values from 1,000 Monte Carlo runs that we conducted with the shadowing of 95% confidence intervals. So as you can see in this purple, there are significant drops in trophic levels, and I have highlighted here to see it a little bit better. And so that's in 1970, 1980, and 2010. And these modeled lines are the IPSL 2.6 and 8.5, both what we called the all parameters and the salinity only models. So next we have biomass, and so similarly we have the years on the x-axis, and then biomass now for tons as kilometers squared. And so now we can see for beluga whales, we have this highlighted here, there's this divergence in the type of Earth system model. So on the top with higher biomass, it's the baseline model, and then the oxygen only models. Whereas at the bottom we have, again, the IPSL 2.6 and 8.5. Anytime there was salinity, we had a much lower biomass. And then here we can see, similarly again, the IPSL, all parameters and salinity only models are creating these large dips in biomass for Arctic and polar cods in the 70s, 80s, and 2010s. And so then our last indicator will be our diversity indicator of Kempton's Q. And so now you can see, again, the IPSL 2.6 and 8.5 are creating very high and very low changes in diversity. And these are, again, in the 70s, the 80s, and the 2010s. So why are we seeing these large changes and sensitivity to the Earth system models with salinity? One possible answer is that the Beaufort Sea is freshening due to climate change and the sea ice anomalies. So in the 1970s, there was a shift from a positive ice anomaly in the beginning of the 70s to a negative ice anomaly in the late 1970s. And so that just means that we had lots of sea ice at the beginning and then considerably less ice at the end of the 1970s. So with this loss of sea ice, we're having weaker stratification, and this allows the warmer Atlantic water masses within the Beaufort to come up and melt the sea ice. And so this is giving us this feedback that's amplifying warming and sea ice. And so with less sea ice, we have weaker stratification, which gives more heat from the Atlantic, causing more sea ice, and the cycle continues. And so furthermore, these ice anomalies in the Beaufort are actually connected with the Greenland Sea, and they're out of phase. So when we have low sea ice within the Beaufort, there's actually high sea ice extent within the Greenland Sea. And these are usually attributed to alternating atmospheric circulation. And this can be from El Niños or Southern Oscillation events, or the Arctic High or the Arctic Oscillation Index. And so this accumulation and release of Arctic freshwater can modify the upper ocean salinity and the subpolar North Atlantic, and thus influencing the ocean stratification, deep water formation, and oceanic circulation. And so actually between 1965 and 1995, the Arctic Ocean exported a large amount of freshwater as ice through the Fram Strait and the North Atlantic. So this freshwater from the Arctic contributed to half the total freshwater in the North Atlantic during the Great Salinity Anomaly, which was a decade-long low salinity event in the 1970s. And there were other similar salinity anomalies that were reported in the 80s and the 2010s. And so in recent years, the Beaufort Sea has actually had this unprecedented sea level rise due to an increase in freshwater in the upper ocean. And this has been attributed to sea ice melt, precipitation and runoff, and changes in atmospheric circulation, allowing for the freshwater in the Beaufort Sea to accumulate. And so this rate of freshening in the Beaufort Sea is actually nearly 4 times that as the Great Salinity Anomaly from the North Atlantic in the 1970s, and has been slowing ocean circulation globally and the seasonal cycles of production. So to conclude, during this portion of my thesis, we found that the Beaufort Sea Shelf ecopath with ecosim model is very sensitive to the changes in salinity, and we've begun to explore some of the historical changes and their connections to sea ice anomalies, atmospheric and oceanic circulation, and freshwater events. These models and outputs can provide a baseline or a reference point for these future projections that we have planned for the Beaufort Sea Shelf, and can advise for management and adaptation measures on the climate variables. And this is also important for cold or warm water adapted species thresholds and any early detection of ecosystem-wide changes or shifts. So thank you very much, and now I'm available to answer any questions.
Thank you, Kristen. Very nice presentation. We have time for a few questions. Yeah. Please come up here and use the microphone.
Hi, thank you very much. Nice talk. Freshening event in Beaufort Gyre is mainly controlled by the Arctic oscillation. Then those are CMIP model, natural variability of the Arctic oscillation index, different from model to model. So there is no reason that at the same timing freshening event happens. So rather than seeing that by years, categorized by those Arctic oscillation index would be more informative to analyze.
I will do that, thank you.
We have time for a few more questions, please.
Hi, that was nice. I'm curious about the model estimates for trophic position. They dip down to 1, I believe,
yes,
for the cod at some point. So do you think that that's real, or do you think that that's just a relative measure of change in trophic position?
It's possible it's just a relative change. I have also looked at, so these were the mean values from those 1,000 Monte Carlo projections, but I also have the values from the best fitting values as well. And so the idea is to compare the mean versus the best fit and seeing what actually is maybe happening when the ecopath model is trying to reduce the number of sums of squares for this as well.
We have time for even another short question. I was then curious about the rate of change. Is that something that you've looked at with the ice, and you say that there's erosion with heating from below? Have you looked at the rate of change?
I will be looking at it very shortly. Unfortunately, it's all very preliminary results. I was still making results last week, so this is a nice opportunity to present and get ideas for the discussion within the paper as well.
Thank you.
Thank you.
So we will go on to the next presenter.
Okay, welcome back, everybody. I hope you enjoyed your lunch. Next presenter is Ben Laurel, and he will talk about changes in overwintering success for juvenile cod species in the Arctic. Please, Ben.
Great. Thank you very much, and thanks for coming back after lunch. So we're going to move into some familiar slides here of climate change and species redistributions and just changing population dynamics. I'm primarily going to be talking about the North Pacific, but I think these processes apply to our other oceans here in the Atlantic as well. Pacific cod is a species I've been studying for about 20 years in the Alaska region, and during the marine heat wave, they experienced an 80% loss in biomass over about a 3-year period. And coupled with those changes were these large redistributions of abundance, both moving north, as we would probably predict, to see colder waters, but also west, where there is also cooler waters. And we've seen a lot of species distribution models and thinking about how we're looking at these from an adult perspective, but we have to remember that population dynamics are generally being determined at early life stages. And of course, Johan Hjort identifying a series of critical periods. What I'm going to talk to you about is an emerging critical period during the winter, something that we've not really thought about as much in the context of spring and summer processes. And when we have changes in distribution of fish, we have two things going on. We have fish that are moving on the leading edge towards those poles, are facing challenges in the winter in form of freezing and longer winter duration. Oops. And on the southern portion, we have on the rear edge here these new metabolic demands, warming winters, loss of seasonality. And there's different processes occurring in each, but often it's bioenergetic processes that govern overwintering success. And we heard from our plenary, there's other challenges, especially as fish move into the Arctic, facing toxins. And we've demonstrated that small amounts of toxin, PAHs from oil, can lead to disruptions in lipid metabolism. So we have from long-term exposure or short-term exposure having long-term impacts on the fish, with, in this case with polar cod, a 30% reduction in the lipid content leading into winter. So what does this mean? And so undoubtedly things are changing. We know things are changing. How are they changing? What are those processes? And can we use processes maybe by reducing some variables and then use those to reparameterize some of the models that we already have. Overwintering ecology, I indicated, was understudied, and it is not new. It's been understudied and recognized. Every 15 years there's a paper that comes out that says, hey, we should go out in the winter and look at this. And of course, it's warm in your office during this that period, so most people are not doing that. They're analyzing their data. And so it's not a new knowledge gap. We know about this. So what can we do? Well, we can borrow from assumptions, and a lot of those assumptions and sort of frameworks that we have for overwintering success in that first year of life comes from freshwater literature. And that's heavily dominated by size-dependent processes. If fish get to a certain size, a critical threshold, they'll have a higher likelihood of survival through the winter. And that's both a consequence of being able to carry more energetic fuel in your tank, as well as standard metabolic rates tend to decrease with increasing body size at this life stage. But when you look at it in marine systems, we don't find those effects. It's been looked at pretty well in Newfoundland now. There's a lot of size-independent overwintering success, as well as in the Bering Sea. We know that there's more of a lipid story here about energy, and that's independent of size. So we need to look harder. We need to look more carefully at this, and probably on a species-by-species basis. One hopeful story, perhaps, is we've heard about the temperature size rule. We know that fish that are smaller in that first year of life actually get a slight advantage with warming. They should be bigger, and that maybe is an advantage to winter success. And we actually see this in our Pacific cod populations in the Gulf of Alaska. Over 4 decades of watching ichthyoplankton in the system, they're getting larger and larger with warming, 50 to 60% larger. And our juveniles are also following that over 2 decades of observations. These fish are not the same fish that they used to be going into winter. And coupled with this is also changes in the size structure within cohorts. Again, in the case of Pacific cod, we had these really unimodal, they're single batch spawners, and they have really tight size distributions. That's also changing. They're widening out. We have now a bimodal size structure, and we can talk later about why that is, but it's resulting in new survival trajectories for these fish. So I had several questions leading into this talk, and I'm going to talk about a recent experiment that was just completed. We've been investigating these processes in the lab with different cod species, and I'm going to talk to you about a recent one. And really was whether or not we can actually forecast winter success based on the summer data that we already have. We catch fish in the summer typically, we have size data, we have abundance data. And then we asked whether or not feeding conditions and thermal experiences in the fall reshuffle the deck. Do they actually sort of disrupt these predictions by different scenarios? And then what can we use other than those summer demographic information? What else can we use to sort of predict survival? For example, can we measure fish condition? And finally, asking a broader question, looking at seasons, not just at a single time point, like a winter metric of maybe temperature. What about looking at fall and winter together? Do these amplify or buffer these effects? So I'm going to talk to you about this experiment. This is where I conduct my research here at the Hatfield Marine Science Center. I go up to Alaska, we collect live fish, we put them on airplanes, they get shipped back. And we took the 2021 cohort that we collected in Kodiak, Alaska. I brought several hundred of these fish back, and we put individual tags in these fish, and then we held them for a fall and a winter phase of an experiment. This is the general schematic here. So these fish, again, were individually tagged, and then put under 2 different thermal treatments, 7 degrees, which was the historical fall experience, and then during the marine heat wave, they experienced these 10-degree falls. Within there, we had replicate tanks where they had low food conditions or high food conditions, so about a 1% to 5% body weight per day. This is a 6-week fall phase experiment, 2x2 kind of temperature food. At 6 weeks, they were sampled for lipids to see how they were performing, and then they were reshuffled and put into a series of other tanks of winter temperatures. These were under starvation conditions, so food was held constant, but we knew the individual fish that were here, what they experienced here. So the first question, does size predict survival into the winter? And this is overwintering days, they would be able to survive these fish on the y-axis and the x-axis is size. And 2 snapshots are what we started in the experiment. So this would be our summer look, and then a late fall look right before they went to winter. And the story here is that size doesn't work, at least not from our summer data. It's a very weak predictor of success. But as you get closer to the winter, if we had a fall look, it's a slight improvement, but a lot of noise, a lot of variation in this relationship. When we add winter information, does that help? So if we add winter temperatures here, staggered from top to bottom, these are the left and right sort of summer and fall look of size dependence of survival, starting with colder winters on the top, moving down to warmer winters down here. The overall effect is that within these relationships are not really improving. We see that late size measurements are better at predicting. We don't really have a consistent winter pattern when we look within this. It's really not adding the information that we thought it would. So let's make a more complex model and let's stop looking at size. And this is recent growth. This is how fast the fish were growing through that fall period. So that's 6 weeks of growth for individuals, and on the x-axis, and then again overwintering days, how long they survived. Here we see a really interesting pattern. We get almost 50% of explained variation just using growth, independent of knowing anything about fall or winter, just how fast those individuals are growing. And you see that it's asymptotic, so it works up to a point. When we add more to that model, we see what's controlling growth and how it's working. Fish that are fed more are growing faster, no surprise there. And you can see how that model works. So the blue is just showing the low food rations, and the red is showing the high food rations. Let's add some more information. So this is the temperature story. So where does temperature in the fall matter for overwintering success? It really only matters under these warm falls. So warm falls, low food, lower survival in the winter, but growth is really predictive. Again, not predictive here. Fish are growing just generally, are surviving pretty well through the winter, and this fall information is not really informative. And finally, where does winter temperature become important? Here on the left panel, we have for the high food treatments that the winter temperatures here for each line, we see that there's this temperature effect. So if you're really growing really well, your fate's not determined until you get winter temperature comes into place. So that's where really good conditioned fish, you should be looking at winter temperature. And as you expect, cooler temperatures let these fish survive longer. Under these low productivity falls, it doesn't really matter. Your fate is sort of sealed. If you're performing well under those low food conditions, then that's about it. The winter temperature doesn't really give you much more help. So it's a lot here, a lot of three-way interactions to consider, but it shows you that these bioenergetic processes are not really that simple. So why is growth a good predictor? Well, it tends to trend with lipid content, lipid density. And there's other theory that this would be a trade-off in lipid and growth, but growth and lipids are walk pretty hand in hand, whereas size doesn't. It's very independent. So size is not the condition metric that we should be looking at. Measure lipids if you can. If you can't, maybe we should be looking at recent growth. So I kind of rushed through a lot of slides there, but just to take home conclusions here and readdress the questions, can overwintering success be predicted from summer observations? And the answer, unfortunately, and this is not something managers want to really hear, but it's not really. The data that we have on hand is pretty insufficient unless we add in fall and winter information. Unfortunately, one of those components of fall information is not physics or something easily measured, it's the food. To what degree does feeding and thermal experience in the fall impact winter survival? Again, temperature here, at least within the ranges that we explored in this study, is not the magic bullet. It's the food. And food and temperature can interact, but food first, temperature second. Do condition metrics based on growth and lipid energy work? And yes, I hope the understanding is that yes, we should be looking at this, less focused on size. And then finally, will warmer thermal regimes, these warm falls coupled with warmer winters, will this amplify overwintering mortality? And this one is, I was kind of guessing it would, but it really is sort of depends. Again, the fall temperature story is less of the importance. It's about fall food. Temperature is important, and certainly warmer winters are not necessarily good. They certainly burn through your metabolic reserves faster. So it's another one of these depends. And finally, I'll end with some management suggestions. We need to improve our fall observation networks. It's just a simple fact. It's not really, shouldn't apologize for the fact that we just don't have the data to answer some of these questions. And we can try to become creative how to do that remotely, but I think we have to increase our observations. And coupled with that is develop modeling frameworks that we can track these fish. So if we get these last looks, we don't have to go out into the winter, but we can parameterize the last look that we have with that information and look at energy loss through that period. And I'm a big supporter of individual-based models that are able to do that and look at those survival trajectories. And then finally, I think this reprioritization. We spent a lot of time on what can we do with the summer fish that we have? Well, let's measure them for a condition. In the case of age 0 fish, I think it's the wrong time of year. I think we should be thinking bumping that down, or at least if you have to sort of prioritize where you want to put your effort, you would probably shift that later in the year. So hopefully that's good, some good discussion for later. I'd like to certainly thank this long list of collaborators and important people, both in the field and the lab, that make these experiments happen. Thanks.
Thanks a lot for a great presentation. We don't have time for questions now, but we have 3 presentations coming, 2 more, and then we have 15 minutes for discussion. So please keep your questions for a little bit later. And our next presenter is Silvana Birchenough, talking about pollution in the Arctic Ocean, overview of multiple pressures and implications for, and I don't have the rest of the title on my phone.
I could say it, don't worry. An overview of multiple pressures and implications for ecosystem services. Thank you, Mette. That's great, thank you. Good afternoon, everybody. I'm Silvana Birchenough. This is a wider view of an evaluation, something we're trying to do. Many collaborators, I'm just trying to bring it all together. My colleague actually who led the study is on maternity leave at the moment, so I said to her I will try to do fair justice to her work. So today I want to just talk to you about one aspect of this work. As you see here, there's many collaborators from Germany, from the UK, and the idea is to bring our understanding, but also to bring the context of what is it that we really have available, so which tools we have available, so a toolbox approach, but also tease out where do we have gaps, what are the kind of things that we need to invest more to make sure that we get a full picture of the types of challenges we're dealing with. I was really pleased this morning about the keynote because it said a lot of the scene of the work that I was going to talk to you today, so that's really nice, thank you. So first of all, the Arctic, just want to really refresh you. I know I'm preaching to the converters here because I know that all of you have an interest. But in reality, we have a lot of level of observations, but also models that demonstrate there is a lot of perturbations, a lot of changes in the Arctic, not only in the physical, but also on the biological systems of the Arctic. In terms of the Arctic species itself and ecosystems, we know that we have a lot of pressures from human activities. We know from this morning again the mixture of pollutants, but also climate change, and also, for example, fishing activities, which are creating constant perturbations to some of these ecosystems and the species. We know also that there is a lot of effort in terms of international policies that are really trying to bring some of the available knowledge, but also supporting the sustainable exploration and exploitation of these resources. So we're not alone. People are trying to really bring things together. But also we know that there is a balancing act in terms of human well-being, but also environmental protection. So there is a bit of a trade-off as well in the system itself. We know as well that there are combined impacts of human activities. We know that we have some challenges in terms of data, but also we know from this morning again, and thank you for the introductory keynote, the pollutants are also causing a lot of challenges, and some of them we're trying to understand and tease out some of the effects, but in some cases there's still a lot of information we just don't know, so we really have to bring some of this knowledge together. So again, we know that from our discussions with our group and our consortium, we know that there is some limited understanding, but also we know that there are specific policies, single policies that are really trying to bring these ecosystem level actions. So one thing to highlight, as I said, this was an effort funded by the UK government under a project called ACEPAC. We're trying to really understand the effects of ice stressors and pollutants in the Arctic cryosphere. But also we have, this is a larger project, and I should say that because this is one aspect of the project, which I thought it would be interesting to bring it all together in a larger scale. But we looked at a lot of the magnitude of the timing of exposures of contaminants to sentinel, but also ice-associated organisms, but also the effects of the lower marine food web relative to the timing and availability of key nutrients, and this is like the nitrogen and phosphorus as well. So today, as I said, this is a larger effort, so I'm only going to concentrate on the work package 4, which again is bringing the multiple stressors and the repercussions for services. And we have tried to take a stock of several tools which enable us to not only understand, but also tease out where we can actually make some progress. So the first part of the work is our approach. We try to bring all of the tools available. We did a lot of synthesis and a lot of screening in terms of what kind of impacts of pollutants we understand and we know, but also what are the actual pressures in the Arctic ecosystems at that level, but also taking it a little bit further in terms of which services they are going to be, provision of services, which ones are going to be impacted, which ones are going to be able to continue, but there's going to be some trade-offs, if you like. But also trying to come up from the angle that things are not going to stop because there is a challenge, there is an issue. So how can we bring some of these recommendations to advance some of this understanding, but also inform environmental practices? Because again, it has to be a level of, I guess, a trade-off as well. So in that regard, we bring some of the understanding not only on the practices, but also on the regulatory processes to say, well, this is the kind of things that perhaps could be aligned better or further integrated better, or some of the things, for example, could actually make bigger contributions to some of our decision-making processes. So we looked at some of the pressures in terms of what are the concerns in terms of the specifics, so what are the very localized levels of pressures, and I'll give you examples how we looked at this. But also in terms of the regional level, because we know that when you deal with multiple stresses, things are happening as we looked into the system, others are very localized, but also there are local communities that are relying on some of these resources. So therefore, we need to really understand the whole of the suite of the biological, the cultural, but also the economic side of it, and what does it really mean. So, don't mean to scare you, this is our overview in terms of looking at pollution. As you see here from the diagram, we're starting to really look at what are the multiple agents here, from the chemical to microplastic, climate change, of course, invasive habitat destruction. Multiple receptors, we looked at the human, but also the ecological. We looked at the multiple chemical exposures pathways, so as you see here, the transport, the multimedia, intermediate levels, but also what are the exposure routes, what are the things that we actually understand. We heard a lot about biomagnification this morning. And also multiple endpoints, what is it? Is it growth, is it community, is it function, is it overall biodiversity? So the complexity of multiple pressures in terms of risk management, what are the things in terms of little from starting from the very temporal to the biological scale to the effect size. So trying to bring everything together into the wider framework of our understanding. So as you see here, we use the UK National Ecosystem Assessment, and we're trying to bring this to the understanding of what we know. So we translate, for example, 4 types of ecosystem services, starting from the supportive, cultural, provisional, and regulatory. And we're starting to really tease out in terms of intermediate ecosystem services, the final ecosystem services, and what does it really mean in terms of goods and benefits. So as we're starting to really bring all of the details, we bring it all into the Arctic ecosystem overview. So we have a long list of tables and matrices and things, you name it, we have it. And the idea here was to actually bring, for example, in this case, we have only taken one snapshot of the pollutants, but also what are the drivers in terms of economic activity. So for example, transport is a very important activity. So what does it really mean in terms of evaluating some of these trade-offs and some of these values and some of these changes? So it took several levels of understanding, because we bring some of the results from the experiments, but also some of the large-scale levels of detail. And we were really trying to rank some of the pressures, but also trying to rank our understanding. It was a very valuable exercise in that regard. There was a lot of list of basic intermediate ecosystem services, but also trying to bring also some of the linkages between them and what does it really mean. So this is just a very overview of how we looked at it step by step. But the idea was that once we brought all of this network of information, we're trying to really bring what is the key ranks of the key pressures, what are the key ranks of the key elements. And from that, we're starting to really tease out some of the key recommendations and key messages of this work. In terms of key messages, of course, we didn't solve everything as we wanted to do it, because there's still some experimental results that were actually being analyzed. But again, I think nevertheless, we made quite really nice progress in terms of looking at what are the kind of things in terms of policy and management that are really directly relating to impacts of multiple stressors and in terms of pollutants. So for example, some of the points here that I summarized, obviously, We came out with a list of ecosystem models. So for example, EcoPath with EcoSim, with EcoTraces. It seems to be a really helpful way to bring the knowledge together. And now we have the data, so we are testing that, but also trying to bring the determinants. What does it really matter? Which ecosystem level responses are going to be more affected than others? So that was really informative, but also really useful to dedicate our efforts how we look at these problems and these challenges. The second part was to look at socioeconomic impacts, and again, because a lot of these sites, a lot of these ecosystems are really providing important services. It was really important to say which ones are going to be the ones that we can actually live without, and which ones are going to be the ones that are going to be challenging and impacted. So the trade-offs was really important. But also as well, in our understanding, there was a lot of documents and there is a lot of advice, which is very high level, but sometimes I think trying to bring those levels of high-level information into specific details, which enable us to not only translate the science, but actually to say, this is the kind of things that we do know and we know well. It's also quite relevant to actually translate what is yet to come that we do need to invest time and effort to understand in full. So also as well, bringing some of the understanding, not only from the science perspective, but actually what does it mean for the communities and the societal needs was also quite important. So not only just the actual pressures levels, but also the interactions and where are the sensitivities of some of these type of assessments. And finally, again, trying to support the discussion and the decision making, the lots of applications in terms of regulatory frameworks. So how can we best bring the science in a meaningful way that can actually really support policy, but also management? So that was another part of our assessment, which it was challenging, because obviously we want to see all of the science being done. But I think it was a useful effort where we actually documented, but also synthesized what is the kind of key things that we do need to know and how, and the time scales for that. So we published this last year. As I said, it was a collaboration between UK and Germany, so we had a lot of exchanges, and all of the summary, all of the tables are in the paper. So happy to talk to you, but it was a really good way, first of all, to understand what we have, so taking stock of the information, but also future recommendations using the science we have, the toolboxes, approaches for management, and obviously policy assessment and application. So I think with that, I'll leave you for that. Thank you very much for listening, and happy to take any comments or questions. Thank you.
Thanks a lot, Silvana. This is really comprehensive work.
Thank you.
We have some minutes for questions, and yeah, we have 4 minutes. So, and because the topic is pollution, we could also have questions for Katrine, talked earlier today. If you have any.
Yes, join us, please.
Frode?
Yes, of course. Yeah.
Specifically, how will you continue this particular project? Will this be ongoing into another project? Could you say something about that?
Of course, yeah. So in the project itself, as I said, we had it's a toolbox approach where we listed a lot of information. One of the biggest challenges, and it's something that we will continue a collaboration through experiments. CIFAS is aligned with the University of East Anglia, and we have an Arctic chamber where we can actually freeze ice and then put species and communities there and incubate them. So one thing to continue from this work is to mix pollutants with microplastics. So that's something that we're very keen to continue. So we have put a grant application to expand on this work. So that's one thing that we're going to do. The other thing is obviously to test our framework, which again, colleagues in the University of Lancaster have a postdoc. So he will be running the EcoTrace with EcoPath with EcoTraces assessment for the EcoPath work. So we will have data that we'll test on the model as well on an ecosystem level. So 2 things immediately we think we want to continue. Yeah.
Thanks. I also have a question, and we are doing cumulative impact assessments for instance, the Barents Sea, and we really struggle with both finding data on, or finding information and knowledge on the risk of local pollutants from vessels and crews, and also to try to understand the importance of local pollution relative to the long-range pollutants. Can you help us with that?
Yeah, no, of course. So in some cases, we did a lot of synthesis of information and data approaches, but in some cases, the information is not available. So we do have to rely a lot on small-scale experiments with similar type of organisms. But also, the other thing is we use a lot of risk assessments to try to see the likelihood of the risk and understand where the gaps are. So in some cases, there's a lot of reliance on what we know and what is expert judgment. And then we try to validate with people that understand what we're trying to do. Similar thing we've done in the North Sea with cumulative assessments methodology. So the likelihood and try to evaluate it like that.
But for the Arctic Ocean, it's the long-range pollutants that are the most important ones in terms of effect.
Yeah, in some cases, for example, we have gone back to some of the old records. CIFAS had some fisheries time series in the Barents Sea from 1900s, and we have collaborated with other colleagues. So in some cases, it's looking at bioaccumulation if we have that information. In other cases, it's just relying on other experiments which are similar. So it's kind of pseudo-analogs. It's not great, it's not perfect, but it's a way to start to say, again, this is a gap. We need to go back and test it in the laboratory. But what we want to do is go back and do incubations in the ice chamber to see what's happening when the ice actually melts, what happens with the contaminants that actually are released, and then see what is the level of effects on a species.
Okay.
Yeah, but we're not there, unfortunately. We're still trying to do it, yes.
Okay, yes.
Thank you. Yeah, regarding the issue of local sources versus long-range transport, because there is a process now through the Arctic Monitoring and Assessment Program where they are reviewing this and assessing what is the status now. I don't know if to look at the historic perspective, but at least they are assessing what are the major sources at this moment and how do they think it will be in the future. And this is a report that will be, I think, presented later this year. So you could keep an eye on that process. Yeah.
Thank you.
Great, thank you. Because I've seen there are some modeling studies from the Baltic and perhaps also for the North Sea, but I haven't seen anything like that for the Arctic regions. Okay, thanks a lot, Silvana. With this, we move into a 15-minute discussion break because. Some friends meeting up here, that's nice. We have not prepared any specific points for the discussion, but we can also open up for questions to Ben if anyone has. Yes?
Thank you. I have a question for Ben. I think your experimental setup is one of the incredibly valuable ways of looking into what kind of processes determines some sort of output. And one of the things that's really great with these experiments is that you can say something about the individual variability, I presume, since you have single individuals that you test. And when you were looking at growth rates or lipids and then survival in the late fall, I was wondering if one of the common drivers of this could be foraging efficiency or the ability to forage more effectively than your brother or sister or cousin, and if you have looked at that.
Yeah, definitely. That's a good point. And so so we have the individual growth rates, but we're growing fish in groups. These are kind of an unavoidable situation. You can't grow individual cod. They are, that tend to be sort of social. So you don't know the individual consumption rates. And the assumption would be that, there's two. It's either physiological or behavioral. What's driving individual growth rates. So unfortunately, we don't have that. We have, group consumption rates and things. And it's a challenging thing to do, with all these sort of growth studies. And the other challenge is that you can't measure lipids twice, right? That's what Sonic over there told me. It's you have to sacrifice the fish. So, but it's, I think, one of the closer times that we've been able to sort of use growth as a really tight proxy of conditions to measure growth twice. And so we can actually look at the role that, by way of a proxy of growth, look at the role of lipids a lot more closely than we have in the past. But yeah, so I don't know if that answers your question, but there's this individual variation and the mechanisms behind it. There's several that even with like this type of experiment, we can't really address.
Thank you. I also have a question for you, and that is because, again, you show that the lipid content is so important, and we know that lipids are extremely important in the Arctic, and the role of lipids in the systems are changing with the changing species distributions and changing biodiversity. So, so my question is, are we monitoring the right things? I mean, are we able to kind of adapt the monitoring systems to the knowledge that we have?
I think that's a good, just an overall philosophical question. We have the temptation to do everything we can with the data that we have, and that's where way we proceed, and that's often, we have to maximize the data, but also recognizing that, we should be open to change and thinking about the way we do things. So I think this is a broad, philosophical question. I am very envious of the Norwegian observation networks that you have in the Arctic. I mean, that is something that we don't have in Alaska. We get these little snapshots. We go up, but we measure everything. But it's only for a couple years. So we have these individuals. We have, they go through the whole, the party of investigators. So we have tons of information for short periods of time. And you guys seem to have these really wonderful observation networks. So I think there would be some more potential to do some, maybe, I don't know if it's prioritization is the word, but certainly opportunities that I see, I think, in terms of monitoring the prey composition, monitoring the energetic value, prey quality, questions like that, absolutely.
Yes, that was my question, because we have extensive monitoring programs, but it's still about numbers and sizes of fish of different species. So maybe we should start to monitoring other parameters as well.
Yeah, it was very much related to the same thing. I was just wondering whether you have been able to actually test this in the field with measurements to do the ground truthing. As you said, it's only the tough guys that goes out to the field when it's cold. Otherwise, the others are sitting in front of the computers. So please reply.
Yeah, I'm pretty guilty of being one of the fair, but we have cold rooms. It's cold in our lab too. But yeah, as far as testing this in the field, so I think it's it's obviously the last observation that you can get and the first one out. So those are like maybe a spring to winter comparison. And beyond, I was involved with some of that work in Newfoundland with a PhD student, and that's very interesting. And that's been very helpful to just understanding that, just getting away from the size idea. And again, we're not measuring the same individuals in that case. We're measuring the cohort. And you have selective mortality, all these processes that might be governing those patterns. But I think that's so. But if you want to get back to this other question of the individual experience, I think there would be some promise with maybe otoliths or fish that you could look at that recent fall growth experience from age 1 fish that you would capture in the spring. I think that would be a really interesting relationship and linking that into the environmental variables. But again, I think the limiting factors in my experience is just having observational platforms to look at maybe spring and fall of a cohort, get a couple looks. So maybe other people have other thoughts.
Thanks. Any other questions? If not, I will continue. I have a question, and that goes to several of the presenters, because we have, so we have been working on many time series, and these systems that we are monitoring, they have gone through warm periods, and some of them are into colder periods again. You had one time series showing this bimodal distribution of size after a heat wave event. So I was just curious to hear, and also in the Barents Sea, Raoul, with the change in community that the fish community that you were presenting. Now we also have a colder phase again in the Barents Sea, and I was just curious to hear if you have looked into the new situations and to see if the system is kind of going back towards an earlier state, or whether you see some changes. And if I could ask you first, because you show this bimodal graph, and I was really curious about that one.
Yeah, thanks. I don't know if Hillary is here. There she is. She'd probably want to comment on this. This is her PhD thesis about cod through the heat wave, and I'll just say one word is that they haven't come out the same, and that's a multi PI effort to understand what's driving that, and maybe Hillary wants to comment on that.
Sure, I can talk a little bit about that. So what I've been doing with the juveniles is looking at how their growth changes before, during, and between heat waves. I haven't personally looked at anything after 2019, but some others in our group have. And what we found with those 2 years between the heat waves after the first warm blob heat wave that ended in 2016 is that these fish are a lot bigger and a lot older than they were before any heat waves occurred. So we actually see that this shift in about 3 weeks of hatch dates, these fish are older, and there seems to be a greater variability in size. So by the time we get to 2021, where Ben sees the bimodal group, it seems like we have, the smaller group in size and the larger group in size, and that could be due to parentage, potentially some phenotypic adaptation of those fish. But I really do think that the otoliths and the growth is key. With the size, you can see changes in size, but you can't necessarily see if that's driven by growth or by age or by a combination of both. And so I think that that's one of the key pieces for untangling what's happening between these events and what will happen in the future.
Yes. Yeah, thanks, Mette. As you said, both the heat waves and the periods of cooling that we experience in Barents Sea now have been instructive with regard to process understanding. So we're looking at community food web level, and we see that there is a sort of return tendency after the cooling period in the Barents Sea. For example, you see that functional diversity goes down and taxonomic diversity goes down in the Arctic. And that indicates, because of the speed of the responses, that these are driven by behavior, essentially. So it's confirming what we of course, suspected, and that is good. But then when we look at the ways in which the change happens, we see that we're not returning exactly to what we had to begin with. So for the future, when there are periods of cooling or heat waves hitting, we probably are going to experience change in certain areas, for example, the Barents Sea, and the recovery or the return tendency from that are quite difficult to predict. That's something that Bergen, for example, showed in a recent paper on the heat waves effect. And that unpredictability will be a big challenge, I guess, in the coming years.
Thank you, Raoul. Any other? I don't see any hands. Yes, Hjálmar.
Maybe I could, on this recent trend, we had this blue blob in the Atlantic in 2015, went through the world media, when this gyre started to expand again. And then we predicted that now we should have more life, both in Iceland, the Faroes, and the Baltic ecosystems. And I think it has occurred. Many seabird colonies that are really doing much better now. cod is about to come back to the Faroes and so on, and more subarctic zooplankton species. So, and that also ties into what we hear here is climate and temperature. Climate is, I just want some of my vendettas here. The climate is much more than temperature. There are large regions in Iceland, the Faroes, where you cannot see a temperature increase, but we're well into natural variability. That's one thing. And people go beyond temperature, that's fine, but when they go into nutrients, just a question too, because I only see here nitrate and phosphate. But the Atlantic is not nitrate limited, it is silicate limited. I think that's important. And I never see silicate, so that just baffles me why looking at nitrates while the North Atlantic is silicate limited. So that's a question to maybe everybody.
Thank you. Anyone would like to answer that? Why don't we focus on silicate? No? We have time for one more comment. If there aren't any, then I think we just move ahead in the program. I need to put on my glasses. Then we are ready for the last presentation before coffee break. So this is Samuel Rastrick talking about natural analogues of an Arctic and rapid transition, the Analog Art Working Group. Please, Sam.
Yeah, thank you. Yeah, so I'm Sam Rastrick. I work at the Institute of Marine Research here in Bergen. And I'm going to talk to you today about the Natural Analog Art Working Group. This is a working group within ESAS. And what we've really tried to do is look at the advantages and challenges of using natural analogues and natural gradients and mosaics within the environment as those analogues. So this working group came out of a meeting we had at the ESAS Science Conference in 2017. And what we tried to do was really get together a group of people who had worked on natural analogues in other environments, such as CO2 vent systems, for example, get them together and actually try to find out what the advantages and disadvantages were of using these models. Actually do this in terms of trying to find new areas in the Arctic. So trying to move this forward to the Arctic where this hasn't been done so much. And importantly, form this cross-disciplinary group of people, because when we looked at it, we often found that, you know, you're off on a cruise here with the plankton people, you're off on a cruise here with the chemists, I'm here doing my physiology on the intertidal, okay, and bring them all together to understand what these animals are actually experiencing during their life is really difficult, but yet very important. And this came out in ICES. This was really our thoughts from this, and from here we've sort of developed this further. So what are natural analogues when we're talking about them here? We're talking about points in time or space where organisms have had time to adapt or acclimatize naturally as part of this naturally assembled system, which is also changing. So the individual species are changing within the communities as the communities change. And these drivers are at a type and level associated with predicted future change. The most simple analogues, of course, are just simple gradients. A classic example of these is all the work done on CO2 vent systems, where we just have one driver changing across space. And by looking at community here or the community there and how the physiologies are within those communities, we can maybe get a view into what things will look like in the future. Mosaic approaches are more complex systems. This is normally what we have to deal with in the Arctic, and this is where we have lots of intersecting gradients. Okay, and we have sites maybe with low pH and high salinity, or low pH and low salinity, and by finding all these sites, we have to try and pull these factors apart. The disadvantage of this is we often had a lot of confounded variables. The advantage, if we can get it to work, is that we can actually use these gradients to look at multiple stresses, not just individual stresses in the real world. And there's a real need for this work, you see, because we are facing a real problem in climate change research, which I think we're all just sweeping under the rug. And that's that all our models, everything, is basically based on taking an animal out of its environment and putting it in a tank somewhere, and then exposing it to its stresses. And these laboratory experiments are great, but they're difficult to actually, in a meaningful way, go from single up through multiple stresses. We can put multiple stresses on them, but these are not necessarily at a seasonal level. They're not of the natural fluctuations we see in the real world.
moving from species to ecosystems is also very difficult. You've removed that animal from all its competitors, its facilitators, its natural food supply, natural changes in quantity, quality of food, and that's also important. Even within mesocosms, this is very difficult to achieve this natural realism. And of course, the elephant in the room with a lot of our work, particularly with the physiology work I do, is their ability to actually adapt to climate. Physiological plasticity is really all we're looking at in these experiments. As well as just looking at physiological plasticity, we can do some multi-generational experiments, of course, but in the Arctic, this is particularly difficult due to long generation times. If we can take animals where they've had time naturally to already adapt to those conditions, maybe we can learn something about their adaptive capacity. So this has been done, as I've said, in other areas of the world. We have a number of papers looking at how species responses to environmental perturbation. We can actually use these sites with space to validate our models, our DEB models, for example. We're doing this in an aquaculture project at the moment with mussels in Hardanger. We can also look into specific tolerances for winners and the losers, and I'll come to that a bit later. We can look at capacity for adaptation and acclimatization, both, as I was talking about, but also in broad evolutionary time, things like Lilliput effect, which we've studied at vent sites. But importantly, again, we can put these physiological changes in the perspective of community change. So what sort of mosaics do we have in the Arctic? Well, this is Hudson Bay. This is an example where the bedrock on both sides of the bay actually affects the runoff of the water, leading to changes in carbonate chemistry. And these changes in carbonate chemistry are in some ways independent of salinity as well. So we can start to look at salinity changes and to carbonate chemistry changes independently. Mosaic approaches. This is for a few days ago. Some of you may have seen Agnes' talk. Looking at fields in Svalbard and looking again at these gradients in carbonate chemistry and salinity, and how they're driven by water masses, seal depth, glaciers if they terminate on land in the sea, sea ice formation, bedrock composition, primary production, for example. Now, by using these kind of sites, we can start to pull apart the effects of salinity and carbonate chemistry in interesting ways, and that's what we've been trying to do. One piece of work, which was done a bit further south by Tina Kuti within the working group, is this study on wall reefs. So we can look at reefs from different environments around different fjords, and we can look at their depth. And across the depth, we have differences in pH, differences in temperature, carbonate chemistry. And we can start to see what's driving these different species and their distributions. And we can start seeing how temperature is an important driver, but also carbonate chemistry, particularly here, look for Litheria, which is a very important species, is also partly driven by these changes in carbonate chemistry. This is some work from Jorge, our PhD student, part of the EMAN 7 project. He's actually up there at the moment at Hawler Reef. And here we actually do have a vent system. So this is a methane vent system. Methane comes out of the ground, and it actually affects the carbonate chemistry. And what we see is that at the lower pHs, we get more soft corals, more sponges, and less dead coral framework, which of course is an important habitat. What's nice about this actually is quite analogous to these things to what we see at shallow water tropical reefs. We see these same sort of changes, for example, when we do work in Dominica. But what's the physiology driving these changes? Well, we can look at some key species within that environment here, Litheria. And what we can do is we can say, okay, so what's driving this? What in the energetics is changing? We didn't see differences in oxygen uptake here across pH, but we did see differences in cellular energy allocation and electron transport chain. And for those of you who aren't familiar with this is another index of metabolic rate. Okay, oxygen uptake is just one index. This is another index. What's neat about this index, it's by looking at the enzymes involved in metabolism. So we can do this on frozen samples. So you can go off on a cruise, collect frozen samples all across with the chemistry, and then we can analyze them later in the lab. And as you'll see, these techniques have become more important in the future, where we don't have to do these large physiological experiments that take a lot of time. We've done similar things with amphipods. So this is a study which we published a couple of years ago now. And this is Kongsfjorden in Svalbard. And what we have here is this Arctic, true Arctic amphipod, intertidal amphipod Gamma setosis. It's got pan-Arctic distribution. And this distribution is shifting due to another species, which I'll come to in a minute. We found no changes in metabolic rate in terms of oxygen uptake, but we did see again at lower salinities these increased costs of survival, which impact the amount of energy available for growth reproduction. I haven't shown all the cellular energy allocation and reserves and things, I haven't got time to talk about it, but it follows this pattern. Unfortunately, with these sites, this is also following a pH pattern. Okay, and this is one of the big problems of these kind of studies, as I said earlier, of these confounding variables. How do we pull these variables apart? And the problem is we need lots of temporal resolution with the chemistry at the same time and lots of spatial resolution. And to do this, we simply can't use these Q10 experiments and things that take 8 hours or large feeding experiments. We have to use some of these enzyme markers. So this is what we tried. So I went back to Svalbard, and I had a lot of fun going around in a small boat, and we got 19 different sites of water chemistry. And we were actually able to pull apart the effects of salinity and chemistry and pH. So by looking at some of these small bays, for example, we could find these animals in areas where we had low pHs and high salinities, and also high salinities and high pHs, for example. And we actually didn't just look at Gamma setosis, but we also looked at its competitor, Gamma oceanicus. So these are the only 2 species of intertidal amphipods here, and they're both really important for birds, fish in this shallow area. And what's critical as well about these 2 species is, as Atlantification occurs, it's predicted that Oceanicus is taking over. But to a bird, does it really matter? I mean, this is a different question. Does it really matter if it's eating Oceanicus setosis? That's a different question. But what we interestingly found was that in actual fact, Oceanicus was actually being more driven by PCO2 in the percentage it made up of the population of amphipods on the beach. Now, this work had been done before, and they thought that some of this might be to do with salinity gradients, but of course all their work, pH was confounded with salinity. We actually found that pH was far more important in driving the distribution of Gamma oceanicus and Gamma setosis. If we look at the energetics, we can see that actually temperature does drive the energetic stress, if you like, on Gamma setosis. Now, this isn't surprising. It's a true Arctic species. As temperatures increase, it's likely to disappear from the fields. However, the idea of Oceanicus can replace it, that's a problem, because Oceanicus is affected by pH. So as this, what we're getting is Oceanicus is coming in to replace, and they're not able to go to those places because of changes in pH and freshening. And therefore, we end up with this situation where we could end up actually not with Atlantification, but with simplification of the environment. So this is just an example of how we can use some of these studies and how we have been using them within the working group. As I said, they've been used in other areas, and we're starting to do this more in the Arctic now, use them for environmental perturbations, use them to link community changes with the physiology, and really tried to move from single drivers to more multiple drivers. Now, to start with, this was actually needed because we didn't have nice CO2 gradients in the Arctic, but it's actually proved an advantage because we can actually now start to look at more interesting interactions between multiple drivers. But we need to keep going, and we have these biological responses to environmental data, and we need the environmental data at the same time. That's critical. So we know what the animals experienced, and we need high resolution both in time and space of the data, and that needs method development and some of the methods I talked about. And then finally, just a plug, our next meeting is actually in June, so if anybody's interested in this, email me. Thank you.
Thanks a lot. Very interesting. We have time for one question before coffee break. Yes.
I wonder if organisms are inhabiting different depths, right? In which there will be a different collinearity between different parameters. You could potentially pull it apart. So then, for example, temperature at the surface and pH at the bottom will be differentially impacting the organisms. So if we were to split by depth, we could get some of these confounding parameters apart. And then, as with the multiple stressors, you are mostly showing physiological changes and population level changes. But what if we are trying to really go from subcellular, cellular, and so on, measurements at the biological level of organization, and then have different pathways being responsible to different stressors. That could be maybe another way of trying to tease apart a little bit of this endless culinarity, yes.
Yeah, so to the first part, yes, definitely with depth. Not with the amphipods, because they're shallow intertidal, that's where we're looking at them. But with the wall reef, for example, that's actually what was done. We used that depth within the fields to, or rather TINA, to pull apart those effects in the community. In terms of looking at the different pathways, yes, definitely. I mean, this is a kind of broad, when you look at cell energy allocation, you're looking at like a broad idea. It's almost a scope for growth for the cell, right? It's this broad idea. But we've done other work where we've actually been looking at things like GABA shunts and things where you can actually start to look at how perhaps some of these changes in GABA may actually be linked to trying to actually control the Krebs cycle, so you actually slow the Krebs cycle down. So the fact that it's linked to low, there's a lot of debate about the neuro side of it, but as an actual control of metabolism, things like the aniline shunt. So we have started to look at some of those things, and again, they're great because you can do this on frozen samples, right? I do think we need to be really careful, especially when we start doing the omics type approaches, that we really careful what we're asking. You know, we know that transcriptomics, by the time we get to the protein, we can be very different. Even proteins can be refolded. So we do need to be a bit careful with those, that we don't just end up with these diagrams where we know something's changing, but we don't know what. So I agree, but I would say caution with some omics.
Okay, thank you very much, and hands for the very good presenters. So now.
It's a great mic check. Have you seen this one? Yeah. Hey, oh, thank you. We're going to move into the final round of the afternoon for this session. Our first talk is Julia Mason, yes, and I'll let her have the floor.
Do I? Oh, thank you. Thanks everyone for coming back from the coffee break. I'm excited to talk about some modeling needs to anticipate cumulative impacts on future Arctic fisheries. So as we've been hearing all today in this session and all week, the Arctic is rapidly changing, rapidly becoming more accessible to new and expanded human uses, from increased tourism to enhanced military presence. And so there's this potential for novel and, of course, cumulative anthropogenic impacts on top of unprecedented and abrupt ecosystem change. And of these, and this change is very likely to be outpacing governance. And of these emerging human uses, fishing represents a real priority, in part because more so than mineral extraction or shipping, fishing is highly contingent on these very complex, very uncertain ecological dynamics that we've been hearing about. As fish are moving northward, there's this Atlantification or borealization of fish communities with very uncertain and extreme knock-on food web effects. Fishing, of course, itself would compound and contribute to these ecological dynamics. But also, more so than any other natural resource, fishing and marine living resources have been a driver of conflict among Arctic states. And this is predicted to continue into the future as fish are moving into new areas. And this type of conflict is concerning not only because of geopolitical strife, but also because the conflict itself can contribute to cumulative impacts by eroding governance and ecological protections. So for example, about 15 years ago, there were mackerel wars in the Northeast Atlantic when Atlantic mackerel moved into Iceland's economic zone for the first time, along with warming waters. And while there was no bloodshed by humans, there were disputes over quota allocation, which led to extreme overfishing of the stock and breakdowns in negotiations for other species. And this is an example of a conflict where scientific uncertainty itself, in my understanding, played a pretty significant role in entrenching and prolonging the conflict, where we weren't quite sure why the mackerel were moving, how long the duration of the stay would be. And so different parties were able to kind of latch onto or promote assumptions or interpretations of the data that served their strategic interests, consciously or not, but this really served to entrench the conflict and create gridlock such that overfishing continues to this day. The fishery lost its MSC certification in 2019. And actually, just a couple weeks ago, the coastal states once again were unable to reach a deal on quota that was within scientific advice, leading to further sustainability downgrading. And so moving forward, as the rate of change, the magnitude of the uncertainty is only going to be getting bigger, this type of situation is really all going to be all the more common. And so to avoid conflict and to avoid potential big ecological impacts, it's pretty imperative that we have proactive and holistic governance for Arctic ecosystems across these different human sectors that can handle considerable scientific uncertainty. And this is particularly true of the central Arctic Ocean, which is in the areas beyond the economic zones of the Arctic coastal states. This is an area that has historically been inaccessible to fishing due to sea ice cover, but is increasingly opening up. There's pretty widespread climate model agreement that we might see ice-free summers as soon as mid-century. And this is also an area of tremendous scientific uncertainty with big limitations on our ability to monitor data and, of course, processes that are kind of novel and unfolding as we're experiencing them. But fortunately, we do have the groundwork laid for proactive management in the form of the Central Arctic Ocean Fisheries Agreement, which we might have heard about in the session yesterday, which is an agreement among 10 Arctic states that agreed to no commercial fishing in this area for a 16-year moratorium. This went into force in 2021, so that gives us until 2037, which is about coincidental with some of the earliest estimates of ice-free summers. And the idea is that these countries establish a joint scientific research and monitoring program that, alongside national science programs and, very importantly, indigenous and traditional knowledge, would form the basis of decisions whether or not we can commence commercial fishing. And it default extends in 5-year increments unless any party objects. And so this is tremendously exciting. It's a landmark precautionary agreement not to fish until we have some more scientific information. But it's also, really, at the end of the day, quite a short window for science and governance to catch up. This is still about the length of time that we've been struggling to get a handle on one stock with a handful of these countries. And so as this clock is ticking on this now 14-year runway, my co-authors and I thought a good first step might be to kind of take a step back and take stock of what can we say about future fishing in the Arctic high seas with the tools and the models that we have now. And we're focusing, based on kind of our personal backgrounds and interests, on marine ecosystem models, which would be a really key tool for informing proactive and ecosystem-based management of the Arctic high seas by projecting future distribution of fish biomass, maybe overlap with other human use, other sectors, ecosystem impacts of shifting stocks. But we know that in the Arctic Ocean, these ecosystem models are going to be limited by their input data, and we have enormous data gaps in the Central Arctic Ocean, in part due to difficulties in monitoring, but also because there are no fish there yet, so we don't have the data about these processes. We also know that global models perform really poorly for this region. We've seen a lot about the spread in the global climate models due to sea ice dynamics and stratification. The ecosystem models that we're then forcing with these climate models also really struggle. This is from my co-author Andrea Brinden-Buchholz's work, assessing the fish MIP ensemble for its performance across ocean basins, with really extreme variability and model disagreement in the Arctic Ocean compared to the other ocean basins. And while there are really high-quality, downscaled regional models for some regions of the Arctic that can resolve some of these dynamics much more accurately, there aren't any that are integrated across the whole Arctic region that have coverage over the Central Arctic Basin, at least that are integrated that we know of. But if you are working with one or know of one or developing one, we would like to hear from you. So global models are what we have, so global models are what we're going to start with. So we're going to be examining the fish MIP ensemble, the fish model intercomparison project, to illustrate some of this model uncertainty. Maybe you heard more about the fish MIP ensemble in the information session yesterday or in some of the concurrent sessions next door. But this is really the global ensemble of marine ecosystem models. It's the one used in the IPCC assessments and to make figures similar to this one that you might be familiar with, showing this redistribution of global marine biomass over the next century. And I think it is really some of the basis and driver of these narratives of the Arctic as a climate winner for fisheries. A quick overview, there are 9 constituent models that link Earth system and climate model outputs to marine biomass and productivity. They're forced with CMIP6 now. On a monthly basis, on a 1 or ½ degree grid out over the next century. And I just want to emphasize that this was a big undertaking. This is a huge multi-institutional collaboration to standardize and integrate these models that is, I think, this year celebrating its 10th birthday. And so what we're just in the very earliest stages of starting to do is to use this ensemble to start to explore some of the Arctic-specific uncertainties and variability. So we're now taking that same map, but looking from a top-down view. We have Bergen down in the bottom here in North America over on the left. So we can start to look at the projections and the spatial projections of where biomass and fish biomass might be moving, and also start to look at some of the standard deviation, the variability in those projections, and what's driving some of that. So we're hoping to look at maybe some of the geographic and spatial aspects of that variability, where the strongest disagreement is, maybe what's driving some of the differences among the constituent models. And actually, one of our first priorities is to run these simulations or run these projections for the near term during the duration of the moratorium. The code is actually running as we speak, so we'll have results soon, but are actually already seeing some big differences among the constituent models for this area. But from what we've been able to pull so far, it's already pretty clear to us that this available ensemble of marine ecosystem models, MEMs, likely really has too much structural uncertainty. And again, that's the model uncertainty based on how we're parameterizing and the models are handling these processes to be useful for informing fisheries management in the Central Arctic Ocean. We can also start to identify some of the big gaps and barriers. We really think that sea ice is one of them that's a barrier for this region. Only one of the 9 constituent models includes sea ice as an input variable. And is really unlikely that it's at a resolution that's fine enough to resolve some of those sea ice and ice edge dynamics that are really important for determining primary productivity dynamics in the Arctic. And of course, we can take some steps to reduce some of the uncertainty in this with improved data collection and downscaled models, but this 14-year timescale is really unlikely to be enough time. And so what we're hoping to do, and we would love input and feedback from this audience, is start to inventory and prioritize the research and policy needs to be able to improve model performance, prioritize these uncertainties, and to move forward. So of course, there's a lot that we can do to improve model performance and increase what we know by increasing our observational data in the region. The Joint Scientific Research and Monitoring Program through this fisheries agreement is going to be key for coordinating and collating monitoring data. Downscaled regional models that are integrated across the Arctic would be a huge priority, but it's also a huge undertaking financially, institutionally, in terms of the time and computation needed. So this would be something we would want to be starting on right away. Of course, this requires sustained funding and protocols for sharing models. We'd also like to start prioritizing some of those gaps that we can address and fill, potentially by starting with coordinating, putting resources toward getting the sea ice people and the fish people to talk, to be able to improve those dynamics that are driving a lot of the uncertainty. But we also want to, instead of just waiting for more and more and more information, start to look for ways that science can support decision-making frameworks that can operate within uncertainty and proceed despite it. And to link these two, we think it's going to be really critical that the research community is really explicit about the magnitude of these uncertainties, that this is not a situation where science can give us a clear answer, at least not on this time frame. And so instead, we want to think about how we can communicate these risks, be really forthright, and be really conscious about how we're communicating this uncertainty. This is something that's come up in a lot of the different sessions. So one option might be transitioning toward more of a risk management framing. So instead of focusing on the unknowns that can be interpreted as, well, scientists really don't know what's going on, use more of this risk management framing that's about identifying the options that would avoid the worst outcomes, the most undesirable outcomes. But we would really love to continue the discussion with this community about how you're thinking about uncertainty in this region, what you're seeing as the biggest uncertainties and gaps, and how we can communicate this strategically and visualize it so that we can inform proactive management action based on what we do know, instead of allowing what we don't know to drive conflict and gridlock. So we'd love to hear from you, and thank you very much for your time and attention.
Okay, great. Thank you for that talk. We actually have time for a couple questions. before we have to move on? Anybody in the audience? Okay, Frode.
So how do you see the need for running multiple ensembles for each of these ecosystem models and compare? And is that very, do you have the computational resources to do that, or is that a limitation?
At least for the existing fishbit models, the code is publicly accessible, and my co-authors are experts in this model protocol. So it's kind of like our time and effort to run the existing models for this region. But for making integrated downscaled models that can like much more accurately capture some of these mesoscale dynamics and the sea ice edges, that is a great question about who would fund and undertake that effort.
Okay, we have a little bit more time if we want to follow up on that or another question. Yeah.
Yes, first I would just like to thank you for your effort in using the fishmeat models. I look into the Arctic because it's really challenging when the models are pointing in all directions, and I try to do a summary of that for IPCC, and it was really difficult, and especially for me as a non-modeler. So thank you for that. We need some guidance on how to deal with that uncertainty in the assessments. You mentioned that only one of these models includes sea ice dynamics. So what are the consequences of that? Have you given that some thought?
Yeah, like we really lose some of the ensemble benefits when there's only one model including this. And so that's why we were, like, again, just based on this very early stages of looking at that, able to like hone in on that as a major gap, and highlighting this idea of maybe more collaboration between sea ice modelers and fish modelers to be able to at least prioritize that as a step moving forward. Yeah, but that's like, it's a huge gap. That's a big reason why we don't have very good agreement, we think.
Okay, thank you.
Thanks very much.
We'll have to move on to the next talk with Cecilie Hansen. You have the floor.
Thanks. Okay, so I'm Cecilie Hansen. I work here in Bergen at the IMR, at the Ecosystem Processes Group. And today I'm going to talk about work that we've done on looking into cumulative negative impact from human activities in Norwegian waters. And it's work that I've done together with a great group of colleagues, both at IMR but also at other institutions here in Norway. So why do we do this? Every 4th year, the Norwegian government updates the management plans for the Norwegian areas. And that is next year. Ahead of that process, they order some knowledge-based information from us. And that process usually follows 3 steps. For a long while, there has been identified vulnerable and valuable areas in Norwegian waters. And this time it was decided that they needed to be updated and new areas needed to be identified. So that was the first part. I'll call those SVOs because that's the Norwegian short name of them. The second one is to assess the vulnerability of each of the ecosystem components within each of these areas that are 19 of in total, to a range of stresses from sectors that are active in Norwegian waters, such as underwater noise connected to marine mammals, fisheries, bycatch, and those species that are impacted by that. And the third one was to look into the cumulative impact of all the active sectors in Norwegian waters within each of these 19 areas, in addition to on ocean scale, so for the Barents Sea, the Norwegian Sea, and also for the North Sea. To do this, we've used the ODEM framework, so the options for delivering ecosystem-based marine management. And in ODEM, the risk of negative impact is identified as the vulnerability times the overlap, where the overlap is divided into spatial overlap and frequency. So we'll first deal with the vulnerability. So that was the second step of the process. We've identified over 18,000 links between stressors and ecosystem components. Each of them are colored, so from purple, that you can see here, which indicates a positive response, to dark blue, that indicates a high vulnerability for the stressor. And the dots has followed the confidence levels defined by IPCC, from 1 dot that is quite uncertain to 5 dots, which is a very high confidence level. We do have a few that are not assessed. For instance, here, between seabirds and smothering, where there is actually nothing to be found. So we left those out. This table here shows an overall assessment for all of the marine areas. And we could do that for all the stressors except for the climate change, where you have quite different responses if depending on whether or not you're in the North Sea or in the Barents Sea. For instance, we have this example of the mesopelagic fauna, where you can have a slightly positive response if you're in the Norwegian Sea, but if you move to the Barents Sea, you see a slight negative response. If we look into overlap, that is between spatial, so that's divided into 3 very coarse groups of 0 to 5% overlap between the ecosystem component and the stressor. So keep that in mind, it's not the area, it's the ecosystem component. And then we move on to those that have a 5 to 50%, and then finally those that have over 50% overlap. When it comes to time, you have those that have a rare frequency. For instance, surveys that visits an area perhaps once a year. You have those that come occasionally, found in the rehearsal areas of the defense, where they use explosions a couple of times a year. We have those that are quite common, for instance, on the fishing grounds, where the fishing vessels come for months, for some months, but not all months. And finally, we move into that last category of persistent that we've usually found between oil installations and pollutants. So I'll show you 2 contrasting examples. We have this one out here in the middle of the basin, deep basins in the Norwegian Sea. And I'll talk a little bit about this one up here in the coast of Finnmark. And I'm sorry for this text, because I noticed too late that it is in Norwegian, but I'll slowly let it through you. I'm not going to talk about any of the other 17 areas, and I would not recommend doing 19 ODEMs in parallel. So let's deal with these 2. Although they look like the cumulative impact is the same between these 2, you need to be aware that they scale to 1. So the total footprint of human sectors in these 2 areas are very contrasting. What you find out in the Norwegian Sea is that there is little activity, but those that are there are a few shipping tracks. We have a little bit of fishery. There are a couple of oil and gas vessels, not any installations. We are there as scientists because some of these areas were assessed for the development of deep sea mining. And then finally, we have a couple of cruise ships coming through. The main stressors that all of these are connected to is underwater noise. And you need to keep that one in mind, because we're coming back to it. It's one of the troublesome stressors. We also pointed towards contamination and littering that happens time to time from things being let off of ships. They are connected quite equally to all the ecosystem components that we find in this area. So we have fish, we have mammals, whales, we have a couple of seals, and then it's the benthos. The color here on the left indicates the vulnerability to climate change. We have so far not been able to identify a way of putting it into the total calculation, which is why it had to be put on the side. But what we often see is that some of those ecosystem components that have a high vulnerability to quite a lot of the stressors also have a high vulnerability to climate change. If we move on to the north coast of Finnmark, the number of sectors are increasing tremendously. They still have that footprint on the ecosystem components through contamination from oil, contamination, underwater noise. But we also see here coming into through the fishery the abrasion that has a direct impact on the benthic, on the soft bed benthic species. We also see that there is a connection through bycatch of quite a few species. And we have in comparison with the Norwegian Sea, where there are no seabirds identified, we have 4 groups of seabirds here that also have a high vulnerability to climate change. If we move on, we can see that I've selected 3 different colors here as troublesome. First of all, the method gives a great overview. It's a core scale assessment, but it points towards those areas where there is a lot of activity, a lot of sectors, and where there are also high vulnerability among the ecosystem components that are identified as a reason for why this should be a vulnerable and valuable area. However, we do have a couple of pressures that are quite problematic. One of these is the pink one here. That's noise. It comes out as one of the strongest ones. It adds up a lot to the cumulative impact from human activities. And that might be true, but there is also an issue with the overlap between the sector and the pressure because noise spreads. At the same time, we haven't used the resilience option within the ODEM framework. So if you turn off the noise, it's gone. It's not persistent. So that might have helped a bit. The second one that we found problematic is the green one here. That is pollution. That also spreads, and it's quite difficult to, there is also a mismatch there between the activity of the sectors, so the ship vessel tracks, and the pollution they leave. In addition, we've had a lot of discussions with the ministry that handles the shipping traffic, and they point towards numbers of how much pollutant the ships leave out, and it's not much. In the Barents Sea, it's 600 kilos a year. So there is a mismatch here on the footprint that they leave in our total overview and compared to what might be out there. The last one, or one of the other ones, is the coral pink down here. That's the fisheries. And the main challenge that we find with the ODEM framework is that it doesn't allow for intensity, or it doesn't count for intensity. And the fisheries in the Norwegian waters, they are very efficient. So they fish a lot of the fish in a very small time of the year. So they do that in a couple of months, and we're not able to account for that properly. Finally, it's also the number of and choice of your ecosystem components. Because between these SVOs that you see here, you see some of them have a quite high risk for cumulative impact, whereas others have very low. But it also depends on the number of ecosystem components. So some of these areas have a few ecosystem components identified as the reason for why it's called a valuable and vulnerable area, whereas others have a range. And when you sum up, it makes a big deal if there are 4 species or 10. So to summarize, it's a great tool. It gives us an indication on where we need more information, probably of a higher resolution, such that we can go more into depth on where should we be more careful, where can we allow for new sectors to come in, offshore wind or offshore aquaculture, and areas where we should be, perhaps we should try to limit our activities. Thank you.
Thank you, Cecilia. That was really nice overview of that risk assessment, and we have a few minutes for questions. I might open up with one if I don't see a hand right away, but so getting into these things at these finer scale these localized, might be spatially with like an oil rig, or maybe it's, like you say, fishing pressure. Is the ODM construct the same? Is that the best approach to use to look at those sort of finer scale features, or are there other efforts that capture those risks?
I think there would be other tools that would be better, but as a start, the ODM gives a kind of a pointer towards where you should start.
Right.
So, yeah, but I like the approach of ODM that you get kind of these 3 different parts where you have information on the time scale, on the overlap, and the vulnerability. So that gives a lot of information that we should kind of keep, but for smaller scale assessments, there might be other tools that would be better.
Yeah, I mean, that's what I noticed, is that with your approach, you see things that are kind of unintuitive that are emerging, whereas I just think of just the obvious things, like the spawning ground and this sort of thing. So, any other questions? Rhoda?
Thank you for the nice presentation. Just one quick question. It would obviously be fantastic to see how this changes over time, but is that possible? Do you have that type of data to bring out a contrast of how this has been changing?
This was based on 3 years of data, so from 2017 to 2019, to avoid the COVID period, because that has an enormous impact on some of the sectors, for instance, the tourism. So we could take another part of, or another slice of time and do the same exercise. We just need, it takes quite a bit of time. So it's thousands and thousands of lines of Excel sheets that needs to be filled out, but definitely it can be done, yes. And it would be very interesting to see.
Thank you very much. Yeah. We're going to move on to the next talk, to Elliot Sivel. Floor is yours.
Yeah, thank you. So, hello everyone. My name is Elliot Sivel. I'm a postdoc at the School of Marine and Atmospheric Sciences at Stony Brook University in the US. And today I want to present some work I conducted during my PhD at the Institute of Marine Research in Tromsø, here in Norway. And 2 things before I start into my presentation is I'd like to thank my co-authors, Benjamin Planck, Uffe Lindström, and Nigel Yokos. And also I'd like to thank the project The Nansen Legacy for funding this work. So this study focuses on the Barents Sea, and we're trying to look at different drivers and how they interact and their effects on the ecosystem dynamics. And as we saw all during the whole week, one of the major drivers in the Barents Sea and on the ecosystem is temperature, because the Barents Sea is a hotspot, and as you can see on the map, temperature has an average increased by 2 degrees in the Barents Sea. And this had consequences on the ecosystem. The first one is that we observed a significant decline in sea ice coverage in the winter. So here on the map, you can see that between 1979 and 2013, sea ice coverage has decreased significantly, but also the sea ice coverage in the winter has varied a lot over that period. So the red area here shows the variability of sea ice coverage over the period. The other aspect that has also been presented in this session is that it had an impact on the distribution of the species community. And in the Barents Sea, you have 3 different species communities. You have an Atlantic species communities in red here on the maps. You have an Arctic species community on the top in blue on the map, and you have a mixed species communities in yellow. And as you can see here on the map, between 2004 and 2012, which matched an increase in temperature, you also saw that the Arctic species community retreated from the Barents Sea, and the Atlantic community expanded its distribution. The other major driver in the Barents Sea ecosystem is fisheries. And here I have a short example, which is capeline. So here you have the bottom graph is the spawning stock biomass from the 90s up to the year 2023. And the upper one are the catches of capeline in the Barents Sea. And you can see that the spawning stock biomass of capelin has varied significantly over that time period. And actually, some of these peaks, as shown in some of the literature, have been correlated to intensive fisheries in the area. One aspect that we need to consider when we are looking into the effects of multiple drivers is that these drivers can interact with each other, and these interactions might actually alter their effect on the ecosystem dynamics. And here we have a nice study that was published in 2018, where the authors have studied the interactions between warming and fisheries. And they show that there is an interaction for most of the species and an effect on most of species dynamics in the Benguela upwelling system, and that these effects are different for the different species. So you don't have a uniform effect for all species in the ecosystem. So what we want to do with the Barents Sea is simply doing a similar work, is to assess and to investigate the effect of temperature and fisheries on the ecosystem dynamics of the Barents Sea. And more specifically, we want to look at the effect on the variability of the dynamics. To do that, we need to use data. So there are two types of data we can use here. Either we have observations, but the problem with observation is that we will not be able to cover all the groups in the ecosystem. So the other option is to use numerical models. And there's one aspect that we need to consider when we're choosing our model, and this aspect is stochasticity. All natural systems are variable, and stochasticity plays an important role. One aspect of stochasticity that we can use, that we need to consider, is that some of the processes that occurs in the system are not well understood, and that to better represent them, we can actually, instead of considering and making assumptions that are not always true about these processes, we can just consider them as stochastic and explore the stochasticity of this process with these processes. And this is an example for recruitment in herring, where the authors here have studied the drivers of recruitment of herring, and the conclusion of this study was that we don't have a clear overview of the drivers of recruitment of herring, and that it might be better to consider it as stochastic rather than making strong assumptions that might result in spurious results. The other aspect is that we might have unexpected events, such as high mortality, that we cannot predict. And finally, if we go a bit further with that, we can have this very unexpected events. So one type of model that we can use to include stochasticity in our simulations is a modeling approach called chance and necessity. And so what is chance and necessity? Chance represents the indeterminacy of ecological processes. For example, prey encounter. It's very difficult to measure prey encounter in the systems. And necessity represents all the physical and biological constraints. So for example, a biomass cannot be negative. It's either 0 or positive. And what we're going to do with these constraints is that we're going to define the state space of the system. And within the state space, we're going to sample randomly the trajectories or the states of the system to reconstruct time series. And this is the part of chance. And so we're going to do that sequentially to build the time series, and we're going to build several thousands of time series to explore the possible variability of the system. And so what you're going to get out, the typical results that we're getting out, is this type of results where we have a lot of different trajectories that are highly variable. You can summarize them, of course, with envelopes, but we need to work not on the envelopes, but rather on the individual trajectories to summarize with statistics what we have in the system. Okay, and so this model is called the non-deterministic network dynamic model, the NDND model, short. And this is what we've used so for our study. So the non-deterministic network dynamic model is a food web model. So the first thing we need to define is a food web topology for the Barents Sea ecosystem. And here we have decided to have a simplified topology with 17 links and 8 groups. So trophic links, and then we added fisheries with 4 links. So The fish, the harvested species are benthos, demersal fish, pelagic fish, and mammals. And so we needed to include a module for fisheries, and how we've included that in the model is that we've included that in the form of harvest control rules. So we went into reports from the ICES, and we've looked into the harvest control rules. And so for fish and benthos, so pelagic fish, demersal fish, and benthos, we just took the values from the harvest control rules. So meaning that when you reach a very low biomass, you just stop fishing. Then between two values of biomass, you increase fishing in a linear way, in a linear manner. And then once you reach a certain biomass, high biomass, you just have a constant fishing mortality. For mammals, there are no harvest control rules defined. So what we did is we took a harvest mortality and we considered them constant over the entire simulations. For temperature, because we are trying to investigate the combined effects of both drivers. For temperature, what we did is we applied the metabolic theory of ecology. So some of the parameters in the model are based on this theory. And so you have here, this T here corresponds to the temperature. So you can estimate your parameter values based on different temperatures. And so when we did the calculation, we estimated that for an increase of 1 degree, you had an increase by 10% in the different metabolic rates. So the next step is, how do we summarize this in an indicator that is understandable and easily communicable with everyone? And for that, we went to an indicator that was published in the 2000, which is temporal stability. And it's pretty simple. It's basically the ratio between the mean biomass of the species divided by the biomass variability, so the variance plus the covariance. Then we had to define different scenarios for fishing and temperature. We had 4 of each. We also included scenarios that were unlikely, like decrease of temperature, decrease of fisheries, fishing mortality. And then we had 2 scenarios where temperature increased and fishing increased, but we also had scenarios where nothing happened and temperature remained constant and fishing business as usual. Okay, so we've crossed all these different scenarios, and we've estimated temporal stability for all the species for each scenario. And the question that we have now is, are the combined effects of fishing and temperature antagonistic, synergistic, or additive? Okay, so this matrix that's now in blue is the matrix of what we have simulated, what we have measured. And we needed to have something to compare to define if we had additive, synergistic, or antagonistic effects. And to do that, what we've done is that we've taken the vectors, so the simulations and the temporal stability for individual drivers effects, so where fishing didn't vary and where temperature didn't vary. And we've re-estimated temporal stability that we would expect if we had additive effects. Okay, so we've done that for different value, and then we've done that for the whole matrix. And so we have in blue a matrix where it's the effect, the combined effects that we've measured through our simulations. And the red matrix is the matrix that we would have if we had additive effects. Okay, and so to avoid, because sometimes the stability were very different from species to species and also within some simulations, we are always comparing the stability measures to the reference scenario, and so we're functioning in terms of anomalies in stability. So how did we define if we had additive, antagonistic, or synergistic effects? So we had, so we are going to have histogram bar plots. So the blue one is the measured effect again, the red one is the additive effects. If both would overlap, we would have additive effects. If we have the measured effects that were higher than the additive effects, we had synergism. If we had the measured effect that were lower than the additive effect, then we would have antagonism. And we had that also on the negative side. Okay, so this is the results we had. This is a bit hard to read, so I'm just going to take one species and zoom in. We have here for demersal fish. So we have here the stability anomaly. These are the different fishing scenario, and this is the temperature scenarios. And the first thing we see here is that stability declined as we increase temperature in our scenarios. The second thing that needs to be mentioned is that stability increased with fishing. So it means that your variability, the variability decreased as you increase fishing. And the other thing is that when temperature was low, we had synergism, and when temperature was high, we had antagonism. Okay. The other results we had is that this kind of pattern was also visible for benthos and pelagic fish, and not for species that were not harvested. So that would impact that fisheries actually structure the effect of climate on the dynamics of the ecosystem. So to summarize, we had combined effects of temperature and fisheries that were synergistic for colder temperatures and antagonistic for warmer temperatures. And we had a negative effect on stability by temperature and a positive effect by fisheries. And finally, we have found that fisheries emphasize the effect of temperature, which really supports the development of ecosystems, so integrated ecosystem management, where we consider multiple drivers and not just fisheries. So if you want more information, this is the paper. It's been published this year. And with that, I'd like to thank you for your attention.
Okay. Thank you, Elliot. We're really a little bit tight on time. If there was really, really quick question, maybe. Otherwise, you could save maybe a question for after the session. Okay. Thank you very much. We're going to move on. I should probably check my sheet. Sorry, I didn't.
Lauren.
I know Lauren, so. It's Lauren Sutton is going to give us a nice talk here, 15 minutes. Thanks.
Hello, I'm Lauren Sutton. I am currently the research coordinator at the Ketchimak Bay National Estuarine Research Reserve, but today I'll be presenting my PhD work from the University of Alaska Fairbanks. Before I get too far, thank you for everyone for sticking it out at the end of this day 4 on this very beautiful day. As someone from a northern climate, I understand how rare these kinds of days are. I'd also like to acknowledge my co-authors, Katrina Eichen, Franz Muter, Remy Pigis, and Claudine Howery. So we'll be talking about functional diversity of epibenthic organisms. So we've been talking about the Arctic kind of all day, but I work in the Alaskan Arctic. So that's this little area highlighted by this red box. And this area is unique in that it's the only Pacific inflow into the Arctic Ocean, with a generally northward flow of water from the Bering Sea onto the Chukchi and Beaufort sea shelves. And while climate change has affected everywhere in the world, this area is particularly affected. To show this, I'm showing time on the x-axis and temperature anomalies on the y-axis. The Arctic is shown in red, and the global temperatures or temperature anomalies are shown in gray. So we can see the Arctic is warming up to 4 times as fast as the rest of the world. Going along with this, we have a continued decline in sea ice. And again, times on the x-axis, sea ice extent is on the y. So these two things, along with other environmental pressures, really affect the Arctic communities. And these shelf areas are known as benthic-dominated systems. And what I mean by that is that due to the timing of sea ice retreat, we have an extraordinary amount of primary production that's mostly ungrazed by pelagic organisms due to a mismatch in timing, and then is transported to the benthos. So this in turn supports a very high benthic biomass, which in turn supports higher trophic levels. So when I say benthic-dominated, this is what I'm referring to. So this is the current regime. With changing environmental conditions, one possible future scenario is a shift from a more benthic-dominated system to a more pelagic-dominated system. In either of these regimes, we assume that the species present have a suite of functional traits that allow them to survive and thrive in these environments. To illustrate that, I've created a cartoon here of a regional functional pool, where each colored shape represents a functional trait. So we assume that regional functional trait pool, that these functional traits must go through multiple filters to create functionally unique communities. If we think about what's going on in the Pacific influence Arctic, thinking about temperature and declining sea ice, we would assume that these changes in environmental filters will then lead to changes in functional composition. And so this is what I looked at. Again, I worked in the Alaskan Arctic, so each of these points represents a different station that was trawled for epibenthic invertebrates and demersal fish, with each color representing a different year and each shape representing a different cruise. I broke this region into 3 different areas, and I'll be referring to these areas kind of over and over again throughout the talk. So the region south of St. Lawrence Island is highlighted by this box, and St. Lawrence Island is one of the bigger islands between Russia and Alaska. I also looked at the Chirikov Basin region, which is just north of St. Lawrence Island, but south of the Bering Strait. Then I also looked at the region, which I called the offshore Chukchi Sea, or it's the northeast Chukchi. So to look at functional composition, or predicted functional composition over time, I used joint species distribution models using a hierarchical modeling of species communities approach. And I chose this because this approach incorporates functional traits, which is pretty unique in the world of modeling, and also pretty new. I used a combination of hindcast environmental variables from our regional Perams model, which I'll get into in a moment, and I used predicted environmental variables from the IPCC's worst case scenario. The second piece of this puzzle is to use functional trait environment relationships, and these were based off of my previous work in this area where I saw different relationships of functional traits either correlated positively or negatively with the environment. And this was all to predict past, present, and future functional composition of these benthic communities. So I used the Pacific Arctic Regional Ocean Modeling System, or PAROMS, and this is a 3-dimensional, temporally explicit ocean model. It models ocean circulation, as well as other environmental variables. But I just focused on temperature, which is shown on the left, and salinity, which is shown on the right. My different regions are shown from left to right, with the most southern region on the left and the most northern region on the right. Furthermore, we broke down our hindcast time into 2 different time frames. We had a colder time frame from 2009 to 2014, and a warmer time frame from 2015 to 19, with the earliest time frame on the top row and the most recent time frame in the second row. Then to use predicted environmental variables, again, we use the most gloomy worst case scenario IPCC output, again for increased temperature, which we see on the left, and decreased salinity, which we see on the right. We used 2 modeled timeframes in the future. So we used a mid-century timeframe from 2040 to 2050, and an end-of-century timeframe from 2090 to 2100. And again, we chose functional traits that had that have shown a previous relationship with hydrography, so either with temperature or salinity. And this was pretty heavily based on my results in the Chukchi and Beaufort Sea. So not necessarily the Bering, but we worked with what we had. So the combination of these traits, which are shown here on the left, create different functional compositions of our different stations. So when I talk about changes in functional composition, I'm talking about the combination of these different traits, or more accurately, the different categories within these traits. So we're sharing 2 main results. The first is that I wanted to see if the categories within these functional traits changed over time. So I've broken these results down into the different regions, with south of St. Lawrence Island on the left going north to the offshore Chukchi Sea. The x-axis represents different time periods, with the earlier time period on the left, later time period on the right. And the y-axis represents the percent of modality. Each of the colors correlate to the different modalities, so you can see if there were any changes in percent modality over time. So first what we see is that temporal changes really varied by region. But I want to draw your attention to a couple results in particular. There was an increase in planktotrophic larval development in both the region south of St. Lawrence Island and the Chirikov Basin region, as well as an increase in calcareous skeleton. That's that dark purple, if you can see that, over time. I bring your attention to these two results because in the Chukchi Sea, it's an area that's increasingly more vulnerable to ocean acidification, so a decrease in calcareous skeleton may in fact be pretty advantageous in this region. Likewise, an increase in planktotrophic larval development in the two Bering Sea regions will also be pretty advantageous, as this region is thought to be basically a seeding community for a future Arctic as borealization continues to occur and species continue to move north. Additionally, I looked at change in functional composition over time. And here, these different colors represent different functional clusters, with the same color representing similar functional composition, or similar combinations of the traits that I showed before. So in our most southern region, we saw a small change, or small predicted change, between the colder time period and most recent time period. And then again, a small change between most recent time period, mid-century, and a large change between the mid-century and end of century time frame. In the Chirikov Basin region, there was a large functional compositional change between the earliest time period and the most recent time period, and this decreased over time. In the offshore Chukchi Sea, we saw, again, a large change in functional composition between earliest time period and most recent time period, small change between most recent and mid-century time frame, and then I predicted a large change in functional composition between mid-century and end of century. So the small change between our most recent time period and mid-century time period, along with the larger change between the earliest time frame and most recent time frame, in both the Chirikov Basin region and the offshore Chukchi Sea region, likely indicates that there's already been a functional shift in these communities in the past. And it also likely indicates that what we're seeing now is what we're likely to see in the coming decades. However, there was a large shift again in the between the mid and end of century timeframes between our most southern and our most northern regions, which might point to a new normal in the offshore Chukchi Sea, or perhaps a larger tipping point in the region south of St. Lawrence Island. So, in conclusion, we did see a functional compositional shift over time, or I should say a predicted functional compositional shift, because this is all model-based. But this really varied by region. And so I think this shows the importance of using regionally specific models, especially in such a wide area. And we also saw that there was likely a functional shift that occurred within the time frame when we looked at between 2009 and 2019, at least in the Chirikov Basin and the offshore Chukchi Sea. But this begs the question of what's going on in the region south of St. Lawrence Island. We would expect to see a functional shift in this area first. If we're thinking about changes in species communities, particularly from a functional perspective, we would expect to see a shift south and then moving north. And that's not what we saw in our model, but perhaps that's because we didn't go back far enough in time. If we looked before 2009, we may have seen a functional shift. And I'd like to really emphasize the usefulness of using a functional approach. Generally, we use a taxonomy to look at changes in biodiversity, but if we include functionality, I think we can glean more about what's going on in the environment. In the future, we're including a less gloomy IPCC outlook. So if we were to take carbon, if we were to take ways to mitigate carbon, as one of our very nice reviewers pointed out, we don't have a ton of variability in this model, so that we're going to add that. And then we're also planning to include carbon chemistry, because now the Perrom's model has pH and aragonite saturation, which was not possible a year ago. And finally, I'm doing this analysis for demersal fish, being contracted through the University of Alaska Fairbanks in this next year to continue my PhD work and see if we can see multi-trophic level patterns. And with that, I'd like to say thank you to all the people that have funded me, and as well as my co-authors. Thanks.
Thank you, Lauren. We have time for one, maybe two questions. I had a question about maybe the velocity of change, and just within the data that you do have, is some of these species able to sort of on an annual basis, you notice trait changes? You could comment on that a little bit, and then I noticed, and then you said something about fish and maybe what you would expect there.
Yeah, well, you know, unlike a lot of these invertebrates, fish move, and so that will create an entirely different aspect to this type of analysis. You know, generally these Arctic species, at least in the Chukchi Sea, there's variability, you know, obviously on a station to station, but not so much within year in the more regional aspect. But the traits really are dependent on who's there, and so your functionality will change the most in response to a regime shift of species.
Okay. Have time for one other quick question? Rhoda?
Thanks for a nice presentation. I was just wondering, the downscaled IPCC climate model, have you seen, it was a bit surprising that you didn't have more temperature increase in that medium-term period. Has it been compared to other IPCC climate models to see if it matches or whether it's an outlier?
Yeah, overall, yes, but we tried to make it more regionally specific, so we actually use the mean. I'm not a modeler, I'm an ecologist, so I've worked, it's been pretty fun to work with oceanographers and oceanography modelers on this for the Perrom's and IPCC stuff. So we went back and forth to use a global model output, but so far the regionally specific is within the ballpark, but you're right, it definitely downplays bigger changes. And I think it would be different if we use maximum temperature. We use mean, and we did not include any variability in that, which, as you know, variability will increase over time.
Great, thank you very much, Lauren.
Thanks.
Our next speaker is Agnes Weiner. Yeah, I hope I got your name.
Yeah, perfect. Good. Hi, everyone. I'm Agnes Weiner. I'm a researcher at NORS here in Bergen and also associated with the Bjerknes Center for Climate Research. We are going to shift gears a little bit now. I want to introduce how we use sedimentary ancient DNA sequencing to go back in time and assess the impact of past environmental changes on Arctic biodiversity. We've heard this quite extensively now, and we know this quite well. I think that the Arctic is changing quite a lot. We have decrease in sea ice cover, increasing temperatures, and a lot of anthropogenic pressures that in one way or another affect Arctic biodiversity. However, there are still a lot of unknowns on how sensitive Arctic communities really are to these current and future changes. We don't really understand very well what the long-term responses of communities are. And this is especially true for microbial communities, so for the lower trophic levels in the marine ecosystem. One way of addressing these issues is to use paleoarchives. So we saw a very nice introduction by the keynote speaker this morning. We can use paleoarchives to assess changes in past populations, community composition, and with this information from the past, eventually we will get a better understanding of how communities may change in the future. With this, paleoarchives play a very crucial role in benchmarking and maintaining biodiversity. And I just want to point out this paper that came out a few years ago. If you're interested in this aspect, check that out.
So far, investigating biodiversity over large timescales in the past has been limited to fossilizing species. But since a few years now, we have a new approach which is called paleogenomics. And when we talk about paleogenomics, we are talking about ancient DNA. And this ancient DNA that can be either preserved in like fossil bones, for example, or it can also be preserved in sediments, in marine sediments, for example, and this is what I will be talking about. So marine communities, when the organisms are alive at any moment, they are shaped by the current climate, environment, and anthropogenic stresses. And then over time, when they die or are eaten or whatever, their DNA eventually sinks to the seafloor, where it gets eventually preserved in the sediment and starts degrading over time. Now, if we go out on a cruise and collect a marine sediment core, we can make use of this DNA that is preserved in this core and can use it as a proxy, so as information to reconstruct past biodiversity. This is usually done in correlation with reconstructions of past environmental conditions. So we use a variety of different proxies or indicators in order to learn about past changes in temperature or sea ice coverage, etc. So overall, if we combine the two, the paleoecology and the paleogenomics, we get a quite comprehensive insight in how the ecosystem looked like in the past. And hopefully we get a better understanding of how sensitive marine biodiversity really is to these changes. There's a few really nice advantages of this approach. First of all, we can directly observe the impact of past environmental changes on marine communities. We can get a very broad taxonomic coverage, so we are not limited anymore to only fossilizing species. And we can assess this over quite large timescales in the past. So currently, the oldest known ancient DNA from marine sediments dates back more than 1 million years, so it's quite extensive. And what we also see is, of course, the result of multiple stressors. So I don't know if it's an advantage or disadvantage. If you want to look at one in particular, it's maybe not so great, but we do get the end result of multiple stresses, of course. Just a bit to the workflow. So the first step is that we collect this paleo archive. So we go out and collect sediment cores. Then we bring this cores to our ancient DNA lab. This is a very important step. There's only very little DNA in the sediment, so we have to be careful to not contaminate it with modern DNA. So we work in a dedicated lab. Then we extract and process this DNA, sequence it, and then comes the bioinformatic analysis to actually make sense and identify what communities were present. And in the end, we integrate this with the other paleo proxies. Good. Today I want to present one project. This is a collaborative project between our lab at North and a lab at IOPAN in Poland. This project is funded by the EEA and Norway grant system, and it's called NIDE, which stands for Nordic Sea Sedimentary Ancient DNA. The overall aim of this project is to use sedimentary ancient DNA to investigate the impacts of Holocene environmental change on past and present biodiversity in the Nordic seas. So we are focusing on this area up here, especially the shelf areas around Svalbard. We've also seen this quite extensively now in the previous talks today, that this is an area that is impacted by changes. There's variability in Atlantic water inflow and therefore variability in temperatures and also sea ice coverage. So our aim is to reconstruct environmental changes over the last 10,000 years and then see how this affected biodiversity. I will be talking about a sediment core that we collected up here in northern Svalbard, just outside the Hinlopen Strait. This is a 5-meter long gravity core, and we did the chronology of this core using carbon dating, and it dates back to approximately 10,000 years, so it is a very nice cover of the Holocene. However, I have to point out that unfortunately the top of the core was not extremely well preserved, so we don't have the most recent past, so keep that in mind, please. The first thing we did is, after the dating, we did the paleoecological reconstructions. So we used a range of different biomarkers. We did magnesium, calcium measurements, and carbon measurements in the sediment of the core. And then, of course, the paleogenomics part. And here, just for people who are maybe into environmental DNA sequencing, we are using a so-called meta-barcoding approach. So we amplify one specific marker gene. And in this case here, that's the SSU rDNA, and in particular the V9 region. This just means it is a marker that it's very broad. So we get a pretty wide coverage of eukaryotes, and in particular microbial eukaryotes. Good. So just looking at the results of the paleoecology. Here on the side, this is the length of the core, the 5 meters. Then here you have the age model of this core going back to almost 10,000 years. And then here, after the keynote talk, you're also familiar with these kind of time series. We reconstructed temperature. So we use alkanons to reconstruct the temperature. And so here, this is the oldest part, 10,000 years back, and then going more to the modern part. And please, for now, only look at the relative changes and the trend, not so much at the absolute values. They are not quite correct yet. I think we have to go back and check that. But you see that in the early Holocene, we have quite a lot of variability. And then starting from like 5.5 thousand years, we have a slight decrease in temperature. The next thing we did is that we used the biomarker IP25 to reconstruct sea ice conditions. And let's just focus on this one here. This is the measurements of this IP25. And we see that we have a slight increase in IP25. This means that there is an increase in seasonal sea ice cover over time. We also reconstructed productivity, also based on alkanon biomarkers. And again, here we see that we have from the 10,000 years, then towards the more recent past, we have an increase in productivity. Carbon burial, relatively, the organic carbon, relatively stable over time, eventually also a slight decrease, but really not a lot of changes. Overall, it is a fairly stable time period. The Holocene is not really characterized by any major changes. This is not surprising, but we do see here this slight change here around 5.5 thousand years. And this could be associated that we had the Holocene thermal optimum down here, and which was like a warmer period, and then we get out into a more colder period again. And this time period, the Holocene thermal optimum, could therefore eventually serve as like a comparison to changes that we are now seeing in the very recent past. Good. To the paleogenomics, this is really just a very simple gel image. So we extracted DNA from the sediment samples and then amplified it and kind of visualized it. And this is just to show that it's just a few samples from the most modern one to the oldest one. And we were successful in extracting DNA going back 10,000 years. Then, looking at, this is unfortunately only very preliminary data so far. There's still a lot ahead of us. But again, we have the length of the core, and here the age of the core. And this is just the Shannon diversity graph. And as you can see, there's not really a lot of changes going on. We do have two outliers here, but otherwise it looks like the biodiversity or the diversity is quite stable over time. And here, if we do a taxonomic annotation, we see pretty much a similar picture, that it is quite stable over these 10,000 years. However, I want to point out that this is a very high-level taxonomic annotation. So this group, for example, includes diatoms. So once the next step is to really look at more species level, if possible. And then eventually we will see more trends over time. So yes, it's mostly stable, even in oldest samples, and the communities are dominated by diatoms, dinoflagellates, and radiolaria. If we look at this clustering method that really goes down onto more or less the sequence level and groups the samples by how similar they are, we do see that we have like two clusters here. And again, this dates back to the 5.5 thousand years. So there is some reason to assume that this change that we saw in the environmental conditions, that it may be reflected in the biodiversity. But as I said, have to look at it in a bit more detail on the species level. Good. So to summarize, we were looking at the sediment core here quite far up in the Arctic. We do see slight changes in sea surface temperature, productivity, and sea ice cover over the last 10,000 years. We were able to extract ancient DNA from all these layers going back to 10,000 years. And we saw that the diversity is dominated by microalgae. And eventually we have some changes in relation with environmental changes. So the next steps are to assess the biodiversity in more detail. And one thing that we also want to look at, we do have a shorter core from the exactly same site than this long core. And that has a very good preservation of the surface sediment. So this will allow us to assess the last few hundred years in more detail. And then, of course, is the combination of paleoecology and paleogenomics. And yes, with this, I would like to thank my colleagues at NORS and also at IOPAN in Poland and our funding sources. And yes, thank you for your attention.
Thank you, Agnes. We have a just really quick question, if there is one. Yes.
Thank you. Very interesting talk. So my quick question is, you use Alkeno SST to compare with eDNA, and the eDNA could include not only plankton but also deep water plankton and benthos. So I'd like to know your idea how to sort out eDNA into benthos and deep water benthos and plankton, etc.
So yes, we did. I was only showing sea surface temperature here. Now we did also reconstruct bottom water temperature, so that and it shows a similar trend. But yes, we do get plankton and benthos, and there are a variety of different methods to differentiate them. One way is, so there's a really good database on planktic organisms that were collected by the Tara ocean expeditions, which may be familiar for some. And so the assumption right now is that we look at what is found only in the plankton in this huge data set and what is only found in the benthos from these DNA sequences. And then we try to differentiate like this a bit. It's not ideal, of course. Some benthic organisms can appear in the plankton as larvae or whatever, so it's not ideal, but that's the first approach. Yeah.
Okay, thank you very much. We're going to go on now to our last speaker. Lennart Schreiber.
Yes. Okay, thank you. My name is Lennart Schreiber. I'm a PhD student at the University of Copenhagen and the Geological Survey of Denmark and Greenland. And I know it's late, it's the last talk, but I'm really excited to share some of the first results from my PhD with you, and I hope I can convey a little bit of that. So the title is Deciphering Climate Change Impacts on Arctic Marine Mammals Using Sedimentary Ancient DNA. And I would like to start with a schematic of marine ecosystem of the coast of Greenland. And as you can see here, the ice sheet from Greenland extends onto the sea. And we also have seasonal sea ice, and the extent of that depends a lot on the latitude. But in general, we have a very harsh environment, and the species living in this marine ecosystem, they're adapted to these environments. And we know now, we have a quite good understanding of the distribution of marine mammals in the region. And this is just an example for bowhead whales. We have, for example, these distribution maps for summer and winter. And you can see the hotspots here indicated in red. And we have this kind of information also for all of the other endemic marine mammals in the region. And the big question is, of course, how will these species react to changes in the ecosystem? and how will they react to an ecosystem that has a lot less ice, higher temperatures, and all of the other impacts of humans. And to understand this, I would like to take you on a little trip back in time. So here we start with the present on the right, and then the last glacier maximum on the left. And so we can see before around 12,000 years before present, there was a lot more ice, and in general it was a lot cooler. In fact, a large fraction of the northern hemisphere was just covered in ice. But as Agnes already pointed out, there's already a phase. There's also a phase in between the early to mid Holocene, and this phase was actually warmer than today. So it's, or not today, but it's the pre-industrial average. So it's kind of the kind of conditions that we're approaching today. And how to investigate this? It's already a very nice introduction, so I don't need to say too much. But we're using marine sediment cores, and they contain sedimentary ancient DNA, but also other proxy data. And they archive the past, so it's a way of accessing information even if you don't have any fossils. And what's critical is that you can assign the samples from different depths to different ages. And just a quick walkthrough through the process. So we have coring on a research vessel, and the subsampling, sample processing, DNA sequencing, and then finally analysis. So I would like to show you the 4 cores that I'm going to talk about today, and they are all in the northern part of Greenland. And we have one on the northeast coast, one on the northwest coast, and 2 directly in the north. So one of the cores, just to give you an example of what it looks like, we have the different layers here, and as you go from the top, which is the present, to the bottom, you reach an age of approximately 9,000 years. So what we did is we subsampled the core at different depths, and then we investigated the samples for we extracted the DNA from these layers, but also looked at the other proxies, or some of the other people in the group. And the method that I'm using is a little bit different than the method that Agnes explained. It's hybridization capture. It's a method to analyze the DNA. And in principle, it's just a way of isolating the target sequences from the background noise. So what you can see here, are these colorful spaghetti. These are DNA sequences from different organisms, and the yellow one is what we're interested in. So we're using probes in solution that bind to the target. This is the hybridization step. And then we use magnetic beads to kind of capture them and isolate them. And the last step is the purification. So we have probes for all of the endemic Arctic marine mammals. Just a few examples here. We have narwhal, beluga, bearded seal, bowhead whale, and ring seal. And next I will show you some of the results, but I just want to emphasize again that we use the DNA detection and also the information that we have from the microfossil record and the other biomarkers to kind of like integrate all of this and reconstruct the past. So let's see some of the results here. Let me guide you through this figure, which may be a bit complex at first. So we have on the x-axis calibrated years before present, and we have 3 phases. One, the last glacial maximum, so before 12,000 years before present. Then we have a warmer phase, as Agnes already pointed out, between like the early to mid Holocene, and then again we have a colder phase. And on the y-axis we have the different species here, and yes, you can see these bubbles correspond to the number of sequences. This is not species abundance. This is more like a strength of the genetic signal that we find in the sediment. So we don't have any information, just based on this, we don't have any information on the abundance of the species. And so the main findings here are, in the first interval, we don't find any signal of marine mammals in these sediment core, in this location. So this is here on the west coast of Greenland, in Melville Bay. Then, as this warmer phase starts, we start picking up a lot of DNA from these different species, and there are a few that are kind of are special. And they are, for example, minke whale, orca, and harbor seal, and also hooded seal, because we detect them here, here, and here, but this is actually outside of their range right now. So we would expect to find them further south, but not so far north. So this is kind of an indication that during a warmer phase, these species will move north. And the other species, as bowhead whale, narwhal, bearded seal, they are adapted to these cold environments. So they can thrive both through these like warmer phases and colder phases, but also their habitat changes. This is just not as present in this core. So to look at the next core on the East Coast, in general, we detect a little bit less, few species less, but there's also an interesting story here because until 10 years ago, belugas were not present on the East Coast of Greenland. So you have a population in Svalbard, but you don't find belugas on the East Coast. But about 10 years ago, they started showing up on the East Coast. So this is an indication also that this ecosystem is slowly changing, or not slowly, but it is changing. And we detected belugas in the past in a period that was warmer. So this is also reinforcing this change of species distributions. What about the other species? We have hooded seal here. Again, this is not in the species distribution today. And also for harp seal, today this would be kind of the northern border for this species. But we can find DNA from this species in the past in this pretty high north location. Okay, from this core we also have information from lower trophic levels. And these are foraminifer counts, it's relative abundance. The layout of this plot is the same, so going from past to present, and the relatively warm and relatively cold phase indicated with the colors in the back. And you can see that foraminifer species that are sea ice associated were not that present in the first part of the core, but then we see a higher abundance. The same for polar and Arctic species, but for chilled Atlantic, warm Atlantic species, it's kind of the opposite. So this reinforces that this used to be a warmer phase, and then it's a colder phase. And the last two locations, we actually merged the results from two cores because they are quite closely, they are situated, the distance is not that far, and also they cover, when you look at the ages, they cover kind of a consistent, they fill each other's gaps. So for this location, we find, again, narwhal pretty much throughout the whole time period, the same for ringed seal, but again, harbor seal, gray seal, and hooded seal, this is not where they occur today. So this is, again, a signal that if it gets warmer, these species will probably move north. And so I'm already getting to my conclusions. So it's possible to reconstruct species histories using less than 1 gram of sediment. The cetaDNA record also confirms our idea that species move depending on the conditions, that they move north as it gets warmer. And there will be winners and losers. So for example, for the seal species that I pointed out, this may be a positive development, but for narwhal, for example, when they have to move north, their habitat shrinks. So that's just important to keep in mind. And I will just end with 2 questions. One, are the current MPAs appropriate for future distributions? There's a narwhal MPA, for example, and as the narwhals shift the distribution, is this MPA still able to protect them? And the last question, how will this also impact indigenous communities that depend on these species and it's part of their culture, but if the species move, what happens then? So I would just like to thank the 2 groups that I'm working with at KU and at the Geological Survey, and yes, happy to take any questions.
Again, really nice talk and exciting tool. Do we have any questions? Couple, yeah, start here.
Hi, thank you for a very nice talk. I was wondering, it's probably a stupid question from somebody who's not working with genomics, but what's the lifetime of a piece of DNA in the water?
Yeah, so the principal idea is that the DNA in the water sinks to the bottom and then it gets deposited and then, well, it degrades, but there are also some preservation processes happening. So the DNA can bind to certain minerals, and other people at the institute that I'm working at, they're looking at this interaction. And so the oldest DNA from the environment that was isolated is now at 2 million years, and it may be in a few years even more. But so this study with 10,000 years is relatively short even, so it can even.
I was thinking more about the water column. So, you know, could you get seal DNA thousands of kilometers away from where the seal was, or does it degrade so fast it doesn't really matter?
Yeah, so I think that's in general for environmental DNA, that's kind of a thing that you have to keep in mind. But it's definitely an integration over an area. But I think the modern environmental DNA studies are more, they are better at resolving this issue and doing like repeated sampling in different locations and looking at the species distribution to then see how the DNA moves in the water column.
Thank you, Lennart, for a very nice presentation. I was wondering, before 11,000 years, you don't see any mammal, and I was wondering how sure can you be? Is it that there were not there, or is it that the DNA may have not made it to nowadays? So do you have any way to distinguish between?
Yeah, so for this core in particular, we looked also at the sediment composition, and we don't see a significant change between the last DNA that we detected and then the lower levels where we didn't detect any DNA. So just based on the sediment composition, I don't think it's likely that it didn't preserve. But of course, like if you do more and more studies like these, you will understand it better how that process works.
Well, thanks again. That was a great talk and a great group of talks all day. And thanks for everybody for staying on time and respecting everybody else's time for their talks. That's it for this session. Frode, I don't know if you wanted to make some closing remarks at all, or no, I defer, the man of the hour.
Okay, I will just want to thank all speakers and everybody who followed this session. I've heard rumors that there is a possibility to also have a glance at presentations after the conference is done. So you can recap on things that you have noticed, but have been unable to note down on your papers. So with that, I would like to also thank the conveners here, Ben and Mette, and also Frans, who has joined in preparing this session. So thank you.